No, yeah.
I actually uploaded online, but
Doesn't matter.
Yeah.
it's okay.
Is PowerPoint
Sometimes
of PDF?
Doesn't.
PDF.
Yeah.
His PPT is good here.
PPT is okay.
Yeah. You uploaded PDF?
Very good.
No,
Thank you.
PPT is.
Okay.
It's good for me, but because I can learn the
Yeah.
comment, yeah.
Come earlier so to make sure.
Thank you.
Okay,
Very
we
good.
check again. If it is PPT, then it's fine.
Yeah, the PPT files are sometimes in the wrong format, so it could be that some text is shifted up or down, but in general it should be fine. There are also like some fancy animations that won't work, but it should be. It should be. Yeah.
Is this a hybrid session?
Oh, it's not going in? See?
It's just.
So.
Because I have Mac, so that's why I use this type of thing.
Yeah, it's okay.
Yeah.
It's now it's
Hello.
The prospect down, no?
yeah. So
And this is okay.
this.
So if you go to mine,
One sec. Presentation.
no.
Or
I
is it-
think if you see like a
We can sort by modified.
yes. So then the first one. then.
Previous page.
Yeah.
Oh,
There will be more, don't worry. Be more.
Yeah.
Zero.
Yeah.
And
And maybe you can check if it's working well because I have copied from Mac.
okay, yeah. Check this. Slide. Do you have any notes that you?
But I don't need, I think I have only notes on the second slide, but I really don't need them.
Yeah,
But I don't need them.
because it's this is always the presentation. So
Yeah.
this is what the audience.
I don't need the notes here because I made my slides myself, so
Yeah.
I know what I'm presenting.
Okay.
And the question is the pointer.
Yeah.
Is this,
Yeah.
Yeah.
strong enough? And then
Yeah, the microphone is working. I don't know. I think.
Yeah.
But for me, the pointer is too high, so I will maybe.
It's on.
Yeah.
Yeah.
But so
Yes,
But then it's better you go to presentation mode and you switch on the
And
pointer.
then-
And that's easy.
yeah, that's okay. So it will, with this one, I can,
Do you have PowerPoint? This is a PowerPoint person?
yeah.
Yeah.
no problem.
But that's not touch.
There's no touch. Okay.
So I think I can move with this. Oh, yeah.
Yeah.
Yeah, that's
So I see-
And it's not working still.
okay.
That's because this one is 1-4.
Yeah, 8.
So currently is only unmuted 1-1 and 1-2, not 1-1, 1-2, 1-3, 1-4.
Uh-huh.
And if you unmute 1-4, check.
Okay.
There we go.
And this one?
This one, I'm gonna mute this again.
Okay.
Just
I need it later. Okay.
it's down here in the corner.
Okay.
Hello?
All down in the corner. Okay.
Yeah, and this,
It's one of our students, he needs to present.
Or
this is, so this one,
okay.
is this one.
And the headset?
Okay.
Here, hello,
That's it.
hello.
Yeah.
Yes. My name is Thomas Geiser. I'm currently representing the chair of crop science here at the University of Bonn. And I have the pleasure to lead you through this session. And I would like to start with the first presentation. And I would like to invite Ms. Tripathi to give the talk about the lead paper, which is role of biotechnology in advancing African agriculture for planetary health. Please, Ms. Tripathi.
This is the microphone. It's just a lamp.
Okay. Okay, afternoon, everyone. And first of all, I would like to thank Professor Christian for inviting me for this, and is the honor for me to be here and give the lead paper. So I'm going to talk about the role of biotechnology in advancing African agriculture for planetary health. Basically, when it comes to the Africa's agriculture, we are at the crossroads because we have to increase the production, but we have like a sustainability, but we have to also see like, you know, the human wellness and with the environment. So planetary. I think I'm quite loud. Yeah, but I'm pretty loud, I think. Yeah. Yeah, yeah. But maybe the person sitting in the last can confirm if can hear. Okay, then I think I can go ahead. Yeah. So when it comes to the planetary health, I think we have to connect the human well-being with the natural system, like, you know, improving soils, pathogen, and biodiversity. For Africa, actually, there is an urgent need for the sustainable productivity because there are like very much of the food insecurity. About 280 million Africans face hunger. So, but if I see like a global big challenge in front of all of us, then basically the increasing population is a biggest challenge, like how to feed that increasing population. And when it comes to Africa, that challenge is even more because population is really, really growing fast. And in order to actually increase the food demand to feed all those people by 2050, we have to like increase our production like about 50 to 70% more food. But then we have similar land and the similar resources. So that's why it's a challenge. But on the top of it, that the climate change is even affecting that scenario. Climate change is not just a talk. We are already started seeing the negative impact of climate change in many places with the increase in temperature, unpredictable rainfall. And so a changing environment always requires changing action. Okay, so now the question is why we need biotechnology. So with increasing demand for food, but with the limited resources, or I should say maybe the same resources, and we still need to close the yield gap in the staple crops and enhance food production. So that means like we need to use the toolbox, like all the tools available in our toolbox, similar to when our car breaks down, like we don't decide which tools to use, whichever is needed, you need to use. So the breeding innovation, including the gene editing, is actually one of them. And the major advantages of the biotech is that the tools are most of the time are quite precise. So, you know what you are doing in order to do the sustainability, and it reduces reliance on the input, like, like fertilizer, the pesticide. So it reduces on that, and it aligns with the planetary health, with more food, with less land, and the less harmful action. So there are quite a lot of tools for the crop improvement, which one can use in conventional breeding, mutational breeding, transgenesis, and now more moving on into the genome editing, which is more precise and predictable. Each tool has its own advantages and disadvantages, and so that's why we have to like maybe complement each with each other tools. These are some of the examples of the gene edited product. Like there are few in the market, or there are closer to the market. Like one is the high oleic soybean oil, which is in the market in US. These are the tomatoes, gene-edited tomatoes for high GABA, which are in the market in Japan. And then the conscious green, which are like a mustard green as a salad, which is available in several restaurants in US. And then the Vexicon is the one which is actually approved for commercialization, but not in the market. But then there are lot which are in the pipeline. There is quite a lot going on in Africa. If you see this map from Egypt to South Africa, there are a lot of projects, but majority of projects are in Kenya, and then maybe 80% of those projects are actually at IITA, at the Becca Hub platform. So I will give you a few examples what gene editing can do. One is like controlling maize lethal necrosis. So maize lethal necrosis is a big problem in maize. It reduces the yield by 23 to 100%, which is costing about 180 million annually, which is a big cost for African smallholder farmers. Most Kenyan maize varieties are susceptible. So Carl Simit and Kotiva, they came together and they developed the gene-edited maize, which has been approved as non-GM. So that means they can go as a conventional into the field trial. So they are already in the field trial. The second example is the striga-resistant sorghum. So striga is a parasitic weed affecting many of the cereal crops. And one of them is sorghum, but is also a problem in maize. And so Kenyatta University and Kotiva again joined hands and developed the striga-tolerant sorghum by knocking out some of the genes which are involved in the germination of the striga. And currently, these are under evaluation in the field in Western Kenya, again as a conventional crop. Maybe I'll give you more detail from my work on banana. So banana is very important crop for Africa, particularly East Africa, which is also quite big grower of banana because is a staple food. Most of the other part of the world, actually, banana is a dessert crop, is like a fruit crop, but there is a staple food. As you can see down there are some of the banana dishes. I don't know, many of you might have tasted them. But the biggest challenge with banana is that, they are affected with a lot of diseases and pests like fungal diseases, Black Sigatoka, Fusarium, there are bacterial wilt, there are viral diseases, banana bunchy top, banana streak, and then there are pests and diseases, pests like nematodes and weevil. Issue is not that the crop is affected by so many diseases and pests. The issue is that many times when you go to the farmer's field, you will find several of them are coexisting. If you see that the map down there, you don't need to read, but if you see multicolored dots, means like, the a different dot is a different pest pathogen in the same place. And one of the reasons why there is a huge yield gap in banana is basically the diseases and pests. The orange bar is the potential what banana can produce, like cooking type and plantain, but what in Africa the farmers are getting is the purple. So you can see there is a huge gap which we need to close if we want to increase the production. So I'll give you two examples, stories. I think interesting story I will say. One is the banana streak virus. When I joined IIT long back, this was the first project why IIT hired me. And I used RNAi technology and I never got the solution. So, I exited from that project. But, when gene editing, particularly CRISPR-Cas9 technology came in and my lab actually we established the tool for editing banana, I revisited this and now this time I cracked it. So the banana streak virus is a double-stranded DNA virus. So it's a BADNA virus. Basically is very similar to HIV, is a retrovirus. So this virus actually integrates its genome into the plant genome. And then majority of the plantain has this virus already. If you test any plantain, you will always find it. The challenge is that this only integrates into the B genome. Basically, banana has A genome and the B genome, and they are triploid. I think my slides are a bit distorted. I've shown that one and I don't see it now. So it originates from two diploids, AA and BB, and then the cultivated varieties are AA, triple A's or AAB. And this is the problem in only the varieties which has B genome, because this virus integrates at a specific locus in the B genome. And then when it is integrated, plant doesn't show any symptom until the plant gets stressed. The stress can be the tissue culture is a vegetatively propagated crop, but also the environmental change, like, increase in temperature or drought. And that's when you see that the plant starts, it becomes into the episomal form, and then it starts showing the symptoms. So we felt like, okay, what I do, means I started this work in 2014 when there was not much reported on viruses in plants, but there was a paper on HIV, and I felt like, okay, because it's a retrovirus, why can't I borrow the strategy from there? So the strategy was like, the virus has 3 open reading frame. And so, and when it integrated into a multiple form. So, my intention was to actually shred the integrated, when you shred the paper, you cannot bring the paper back, right? So that was my whole idea. So, shred the whole viral genome, which is integrated. So we designed the guide RNA for the targeting all the 3 open reading frame. And then for the third open reading frame, which is like denoted by red here, it encodes all the essential genes which virus needs, for multiplication, for coat protein, and for movement. But then is a polyprotein, and it needs to get cleaved at the aspartic protease in order to make all these proteins functional. So I thought like, okay, if I can make aspartic protease non-functional, indirectly, I'm making all the proteins non-functional. So that was my hypothesis. So I've designed 3 guides targeting all the open reading frames. And target 1 and 2 was like 198 base pair apart. And if the molecular Cs are cut simultaneously, we should have a big deletion of 198 base pair. But then the target 3, which is more important, but because if there was only one guide, it should give indulge. So we put the guides in a one plasmid, and then we delivered them into we use cell suspension. We delivered the cell suspension, generated the edited plants. And then we did the molecular characterization, means we sequence the targets. And we saw like what we were expecting in terms of the mutation. So it was very precise target mutations. And then after that, we did the phenotyping, means like we mimic the drought in the greenhouse. And then you can see the first plant, which is control non-edited, showed the very nice streak. But then majority of the plants didn't show any symptom at all, apart from only 2 plants. And then I was wondering what happened to those 2 plants. And we saw like, okay, they have the mutation in the target 1 and 2, but not at the third target, which was actually more essential for the viral genome. So now we have like a proof of concept that we can inactivate this virus. And now we are actually helping breeders in editing the parents they are using into the breeding program. The second story is where we have targeted more of the endogenous plant genome rather than the bacterial. So that's a bacterial Xanthomonas wilt. And this is a big problem in the Great Lakes region in East Africa, which is also the major producing banana area. Basically, the disease is spread through insects, but then also the planting material and the cutting tools and all those things, also the spread, and the losses can be between 40 to 80% overall. But once the disease is into the field, it spreads like a bushfire. Within a month or 2, the whole field gets wiped off. And all the cultivated varieties are susceptible, apart from the wild type, Musa balbisiana. And then I think once the disease is there, farmer has to uproot all the plants, leave the field fallow for some time before they can plant. So it's also a lot of laborious activities there. So my lab, we studied what's the difference between the wild type banana and the cultivated? What's the molecular mechanism in that? So we did comparative transcriptomic at the early stages of the infection, and we saw lot of differentially expressed genes between the susceptible and the resistant wild type. So what we did, we mapped these ones to the biotic pathway, and that's how we have identified the genes which we can actually play around for the gene editing. So we have identified a lot of susceptibility genes. These susceptibility genes are involved for the disease development, or some of the transporter genes which or the transcription factors which involve as the negative regulator of the defense pathway. So so far, we have tested like more than 12 susceptibility genes individually because we have to test individually to understand how that work. Not all of them gave us the resistance, but the one which are highlighted in brown, these are the ones which gave us the very enhanced resistance. One of them is the early nodulin like gene, Musa inode 3. And basically, the nodulins are involved in symbiosis with the rhizobium in legumes, but these genes are quite functional in non-leguminous plant as well. That means they do play a role in those plants, and then maybe in the nutrition transport. And then when the nutrition transport also help the pathogen to colonize, So we said like, okay, if we can knock down this one of these gene, early nodulin 3, and see what happens, because that was the gene which came up in a transcriptomic, which is like highly activated in the susceptible variety. So first of all, we did like, we did the phylogeny of the whole gene family to understand where the banana gene sits in that family. And then after that, we checked through the qRT-PCR, and we saw like, this gene gets like more than 20-fold activated during early infection with the pathogen infection. So we again developed the guides to knock out this gene, and then we generated the edited plants. And then we did molecular characterization for the targeted mutation. And then after that, we did the phenotyping. And in the phenotyping, we saw very clear enhanced resistance, many even showing complete resistance in comparison to the control, which got completely wilted once we challenged with the bacteria. But then because the genes are also involved in other pathways, so, we checked whether knocking out, we didn't knock out all the early nodulin gene, we knocked out only one of them, but then we checked like for their effect on the plant growth, and we didn't find any negative impact. If you need to more about this, you can also read. This is published in Plant Biotech Journal. The second was very important thing is like Musa pub genes. So these pub genes are basically involved at the first level of defense, which is PTI. And they are like a E3 ubiquitin ligases, and they have role in immunity. But some of them, particularly the U-box E3 ligases, like PUB22, 23, 24, they are negative regulators. So that means when they are activated, they enhance the disease development, means they suppress the plant immunity. In Arabidopsis, there was a little bit, not through CRISPR, but there was a paper to show that, the triple mutant where they have knocked out 22, 23, 24, it has shown some resistance against the fungal pathogen. So, but in our transcriptomic, again, these are the two genes which were highly activated during the infection. So we created the double mutant, and with the simultaneously knocking out PUB22 and 23, and it has really provided us the enhanced resistance. So again, is the same, like, we have mapped whole gene family. We have checked through the qRT-PCR the relative expression of both the genes during infection, and then we design our guides specific to the U-box of these proteins, so that so that is very precise and targeted. And then we develop the edited plants. We sequence. Once we sequenced, then we know, like, through the SNPs and all, how to differentiate between the two genes, even though the guide RNA were targeting both the genes. And we clearly see there was a mutation, and we picked the one which had double mutation, means both the genes have been knocked out. And we did the phenotyping. In this case, actually, we got 100% result. All the events we tested, because it was double mutant, we saw a fantastic result in the greenhouse upon challenging with the bacterial pathogen. No impact on the growth, at least in the greenhouse stages. And then we also check for the reactive oxidative burst, because that's like one of the indicator that, the defense pathway. And we saw the oxidative burst was much higher in our edited events in comparison to the control upon infection with the bacteria. So one another gene we tested was DMR6, which is a downy mildew resistant 6. This is very well characterized susceptibility gene. It has been tested in several crops like tomato. And so we have to look for the ortholog into the banana genome. And we found there were 7 orthologs, but we didn't want to knock out all the orthologs because we also know that this gene is also involved in plant growth. So we carefully picked one ortholog and we knocked out that one ortholog. And then, we have knocked out, and we have also, by this time, we have very robust screening method where we don't need to even pot. We can do it into the tissue culture. And so from where whatever survived, we did screening with the potted plants in the greenhouse. And we saw again with this 100% resistance with no impact on the growth. You might be wondering why we are checking so many genes, because the single gene based resistance actually break down very quickly because as the pathogen evolving so fast. So our intention was actually to knock out multiple genes. I think I didn't, that is very quite preliminary, but now in our lab, we have knocked out 4 susceptibility genes in the same event. So these are like early nodulin, PUP22, 23, and DMR6. Initially, when I was knocking out, I was curious, like, okay, these plants will not become lethal and they will not grow, but in the greenhouse, the plants are growing like any normal plant. So apart from banana, gene editing you can use not only for the disease or pest resistance, you can also use for other things. So in yam, the biggest problem is that people who are from Africa, yam. Yam is a staple food crop in West Africa, but yam is a vine. So farmers have to actually stake them. So that means it also leads to deforestation because they have to cut the wood, but also is quite laborious. And then this crop cannot be mechanized because of that. So we have developed the CRISPR tool for yam, and now we are creating we are changing the architecture of the plant by knocking out some of the genes involved in the gibberellin pathway and making them bushy, like a small bushy structure, so that they can be planted more compact and then they can be harvested mechanized way, and farmer doesn't have to put that much labor to this. We are working on other crops, like for making them more resilient. So one is the striga tolerant maize. And so if we are actually knocking out some of the genes we have identified. We are also working on tomato for making them. So there is this request came from actually the growers. Like, there is a very popular variety in Kenya, a very high yielding, people like the flavor, everything, but then it is susceptible to bacterial wilt. And because at IITA, we have a lot of experience on bacterial wilt resistance. So I think we are developing a bacterial wilt resistance. So it's the same germplasm, which has everything in it, high yielding flavor and everything. And we are just adding the disease resistance. Another problem is the drought tolerance right now. So we are also working on drought tolerance for maize. And basically, biotechnology is becoming a game changer. Currently, there is a problem of the red palm weevil in Middle East, where they grow a lot of date palm. So we have been requested, IITA doesn't work on date palm, but we have been especially requested, particularly myself, to lead one of the work stream to using the biotechnological tools. And so we are using RNAi and CRISPR in order to develop making the crop resilient for the date with the date palm resistance, and also checking at the microbiome intervention. So this is just to show you that the global landscape for the particularly gene editing is changing very fast. All the green countries have the regulatory guidelines in place. And Africa is really, really, really great. I think I should have put Ethiopia as green. So there are 5 countries right now where if you have no transgene integration in them, they are treated as similar to conventional. Europe is changing mind as well. They have gone to the discussion, so which is a good signal. Only like South Africa and New Zealand is changing from next year as well. So it's only South Africa which treat gene edited as GMO. But the issue is still the harmonization of the regulation. Each places the regulatory guidelines are very, very different. As genome editing is new, it requires lot of communication and capacity building. Means I feel honored to say like, oh, we are the only one in the lab who have like, in sub-Saharan Africa, who have like a full pathway, but we don't want to be that way, right? We want many other institutes or the universities coming up with a critical mass of people, because one institute cannot solve all the problem. So at IITA, we are heavily involved in capacity developing through PhD students, through training the scientists, but also bringing journalists to the lab. Seeing is believing. So they come and they see. So upper picture are the journalists who are really inside our growth rooms, seeing like, the plant looks normal. We are not doing something different. And then also the policy makers so that they understand the technology better. So the key take-home messages from this is like, biotechnology offer transformative solutions for food security, climate adaptation, ecosystem sustainability, and equitable agriculture. And gene editing is a quite practical, safe, and scalable tool, is easy to adapt, and is very, very precise. Africa is already pioneering this application, and policy and the social engagements are essential if we really want to see the benefit of these technologies, especially for the smallholder farmers. So at the end, I would like to acknowledge IITA, my plant transformation team, molecular team, and the bioinformatics team. And also our partners from different universities, and then the donor support. If you want to know more, you can find a lot of resources on our website, which we have created recently, called Genome Engineering at iita.org. So thank you very much, and I will be happy to take any question.
Thank you, Ms. Tripathi. Very nice talk. Are there questions? I would like to allow 2, maximum 3 questions. Can you bring we need a microphone so that the audience in the Zoom can also hear your questions.
Thanks for this very inspiring presentation, really very interesting. I was just wondering, maybe but how do you make sure that these crops then also reach the smallholders who are not always even investing in improved seeds and so on, so that you're really reaching the impact that you want?
Mm-hmm. Yeah, that's a very good question. So, IITA is a public, so we develop for the public goods. And rolling out means the seed system. So we work already while we are getting ready. So now these plants are going to the field trials, our bananas, and we are already started working with the national programs in Rwanda and Kenya, and then some tissue culture companies, so that, they are actually accessible to the farmers at no increased cost. First time, definitely, we might have to find out the ways how we can distribute for free, or with subsidized rates. But then the good thing about the crops we are working on, like they don't have to go back to the seed company again to buy, like, the bananas, cassava, yam, vegetatively, so they can get the seeds even from their farmers. So I think we have like a full plan on that. Means 30 minutes were not enough for me to talk about that. But if you want, we can discuss. We have full plan how we can roll out this to the smallholder farmers.
Christian, you had another.
I have a very quick, and thank you very much, Lina. Very interesting talk. As a chocolate lover, I'm asking myself, on your future list, where's cocoa?
Yes, that's a very good question, because I know a lot of cacao is grown, but now I think we also need to see, the CG scenarios are changing, and we are very much as actually the project based. So you have already seen, initially, that I was only working on banana. Our crops are expanding, and one day cacao will come on the list as well.
Last question, lady in 96, number 96. No? Yeah, 96.
Thank you so much, Dr. Lina. This was really enlightenment. So I would like to ask about the banana streak virus. So there's no way to clean this in tissue culture. And all the plants are what you know for now, it's the virus is integrated in the plant. So there's a pipeline to check this in tissue culture already, so that we can already from tissue culture level know what is it integrated or not.
Yeah, so you cannot clean it with the tissue culture. With the meristem tip or all, you cannot clean the streak virus because it's integrated. And reinfection with this edited plants will not be possible because, I told you that is only in the B genome and is a very particular locus on the chromosome 1. And what we have done is we have not removed the complete viral genome from there. So we still left the viral genome so that the site is occupied, but we have shredded it so that it cannot go back into the activation form. But in tissue culture, also we don't see the symptom during the tissue culture. What the symptoms we see is when you wean them in the greenhouse. That's when, because that's the stress. That's when you see the streak symptom.
I have another question if it's still okay. So when
Very small question.
yeah, so when we speak about the bacterial disease, so you knocked out several genes. So that means this the resistant here is recessive resistant, not dominant, right? And when
Yeah, it is a recessive
so
resistant.
this make it more difficult.
Yes, because then you have, and then is a ploidy, like is a triploid. So yes, it is difficult because then you have to knock out the 3 alleles. Yes, because, and it is a recessive resistance.
Thank
Yes.
you very much for this enlightening questions and answers. I would like to invite the next speaker, Ms. Kiboy, to give her talk about the performance and yield stability of maize under a long-term experiment in the subhumid tropics. Please, Ms. Kiboy. You should go as close as possible to it. Yeah. Should work. Is this the correct one? Try.
Okay. So this
Does it
is,
work with the pointer?
no, I think this is
And
PDF.
then you have to use the keyboard
Yeah.
to scroll. It's not in the presenter mode.
Yeah, it's a PDF.
That's why.
Yeah, okay, I have to do it.
Different format. Okay.
Let's see.
Yeah. That's a full screen now.
Yeah, it's okay.
Sorry. Yes, it does. Okay, welcome to this presentation. I'm going to present to you some information on maize performance and yield stability and our long-term experiment that we have been running since 2007 to date in different countries, but I'm gonna focus only in Kenya. So I come from the Research Institute of Organic Agriculture in Switzerland, but then we don't implement the trials by ourselves. We have our partners from ICIPE and CALRO. And the title of my presentation will be Maize Performance and Yield Stability under a long-term experiment in the subhumid tropics. So first, I start with the challenges that the farmers in the tropics face. And what I'm focusing on here is the cereals, particularly maize, which is the main staple crop globally, Sub-Saharan Africa, and in Kenya. And for those of you who may not know more about Kenya, is that maize is a very common dish for the population there, and we eat a meal called ugali. It's purely corn. You use that. But then what has been the challenges with maize production in the tropics specifically? Kenya is the declining soil fertility that is mainly caused by the low use of inputs by the farmers because this is not affordable for them. Also, the infrastructure, that means accessibility of these inputs for the farmers is also a challenge for them. But then again, we recommend to use organic resources. But then the question is, how are these organic resources managed by the farmers in terms of the quality, also the quantity? When do they apply the organic resources? These are the challenges that have led to a decline really of the soil fertility in the tropics. And this leads to low productivity, and it's coupled with monocropping. Season after season, the farmers are only planting maize. Climate change is also one of the challenges that the farmers are facing in the tropics. And this includes frequent droughts, erratic rainfall, and then you have prolonged dry spells within a season, or a change in the cessation or onset of the rainfall. Now, when most of us researchers are working on different crops, the productivity, we mainly focus on the yield, the amount of yield we get from these different farming systems. How about the yield stability? That is the resistance, the resilience of this crop in productivity. And how we define this yield stability is the consistent performance of the crop, be it at high or low yield, across a range of environments, at different external inputs, as farmers are using different external inputs. So we have been implementing different farming systems at different two sites. And so for this study, we were checking on what are the effects of organic and conventional farming systems on crop and temporal yield stability with different external inputs in the long-term experiment. So the long-term experiments I'm talking about, they're established in two sites since 2007 to date, and they're established in a randomized complete block design with 4 farming systems, which I am going to talk about more in the next slide. But generally, we have high input systems that mimic what a commercial farmer does, and the low input systems. Where we have the sites is a place called Chuka and Kandara, and the two have almost the same characteristics. For example, the predominant soils there are the nitty soils. However, in Chuka, we have the humic nitty soils that are better in terms of soil fertility status, while at Kandara site, we have the rhodic nitty soils. So these are the mean annual rainfall that are reported in most of the publications, but we are going to see on what we have been receiving since 2007. And the mean temperature for the region, for the two regions, is mainly 20 degrees Celsius. So we have 4 farming systems, the conventional law and organic law. And here how we determined their law is because we carried out a survey as we were beginning this experiment to see what are farmers doing in the regions where we are implementing these trials. What are the rates that they are using in their fields for different crops? And then for the conventional high and organic high, what are the farmers who produce for the purposes of selling the produce? What amounts of inputs are they using? How are they managing different aspects, for example, the pests and diseases? And are they using irrigation or is purely rain-fed? Most of the farmers in the region where we are working is that they are smallholder farmers and they are mainly doing rain-fed agriculture. But then we do a 3-year crop rotation cycle in the experiments. And at the beginning of each rotation cycle, we plant only maize in all the systems. Whereas in the second, we do the vegetables. And then in the third, as you see, you may, it's baby corn in the high input systems, and then maize, the Z maize in the low input systems. But then our focus here today is maize, because it's all in all the systems throughout every beginning of the rotation cycle. And just to say is that we have bimodal rainfall in the region, whereby we have 2 cropping seasons in a year in the 2 regions. So the first cropping season, we call it the long rain season, that starts from March to June, and we are harvesting around July and August, while the second season starts from October to December, and we harvest in January. So what we observe only in the maize cropping season throughout the experiment is that we have a variation during the seasons and also the years. And the frequency of dry spells of 5 to 10 days dry spell in the region has been quite high since 2007. Maybe I may not show the results of the frequency, but this is published, and you would see that you even have 4 to 3 times when you have a dry spell within a season, and more so the long rain seasons where we are planting maize and where the farmers mainly plant maize. And this also gives evidence of climate variation in the region. But then we come to the results. What have we seen in terms of crop indices? And here we are looking at height and diameter, since these are important agricultural indicators towards monitoring the physiological traits, maturity traits such as grain yield and biomass. So what we observe as expected is that in the high input systems, the height is of course higher than the low input system, but what we see is that towards the later years is where we are having now all the systems not having significant difference, especially more in Chuka, where the soil fertility status is better as compared to the Kandara site. For the low input systems, we see that the organic loop consistently has not been performing well. But remember here, the organic inputs added into this system is also low, as the farmers do. And also the application time has been a challenge for us. Now, in terms of the grain yield, at the beginning, we have the conventional high doing very well. Remember, the conventional high has an integration of the synthetic and the organic inputs. That means that the synthetic inputs, the nutrients are already available when we are planting. The organic inputs require moisture for them to mineralize the nutrients and make it available for the crops, which may not synchronize with the plant demand. But with time, then this has increased, and the organics have now been performing well in the later years. To the subject matter, the maize grain yield stability. Now, despite the high input system consistently performing well in terms of yield, this does not mean that they are also stable in terms of the yield that we have been seeing. So the organic low has been showing stability in terms of the yield, except in Chuka, where we have the conventional high doing better. And this we saw when we have the residue variance being heterogeneous, indicating that these systems have an effect on the yield stability. So in conclusion, what do we say and what do we recommend is that long-term research is important for soil improvement and for soil adaptation to the different changes that we have done. And the organic farming systems, they have the potential to achieve yields that is equal or exceed the conventional farming system. The low input farming system, they show greater yield stability, and they could essentially play a role in mitigation of the risk or the effects that we see from climate change. And this being an area that is very susceptible to climate variability, it is important to consider yield stability alongside the yield performance. And the recommendation is to sustainably enhance maize productivity and stability. Then we need to improve soil fertility through sustainable organic practices. So as I said earlier, we have our partners that we are working with in Kenya, and these are our coin donors who have been with us since 2007 to date, and the long-term experiment still continues. Thank you very much for your attention.
Yeah, thank you. Great talk and very well in time. I forgot to say that please keep your questions until the end of the session, and then we will discuss the last 4 talks together. So May I invite Mr. Fredua to give his talk about evaluating sustainable maize intensification strategies in smallholder farming in northern Ghana.
When? Absent. When you send. So, yeah, this is the update.
We don't have time to change. We have to, you have to use this one.
Okay.
Sorry. Time is too short. I guess it's still okay.
Yeah, I'll manage.
You will manage.
Control.
On showing you results. You know, you have only 10 minutes.
Okay, yeah.
If necessary, keep most of your introduction and focus on the result.
Okay.
Thank you.
Thank you too. My name is Bright and I'm presenting on the topic evaluating sustainable maize intensification strategies in smallholder farming systems in northern Ghana. In order to improve yield, several intensification practices are being done in the region, such as the use of inorganic fertilizer, organic fertilizer, improved varieties, ones that are drought tolerant or short varieties, shift in planting windows, crop rotation, as well as intercropping. However, which sustainable intensification practices are suitable to bring expected sustainability or stability in yield is a question that remains to be answered. In order to help us with this study, we came up with the objective to assess the performance of sustainable intensification practices, namely soil application of inorganic fertilizer or manure, and the combination of this different maturity duration maize varieties, different planting windows, as well as the study to be carried out in 3 different districts. The districts so the it's no full. Are you changing to PDF?
Yeah, this is
Okay.
already PDF.
Okay, so. So I use the arrow?
Is it okay? Yeah.
Yeah, okay.
So,
Okay.
not bottom.
Right. So the study focused on the 3 areas, Tolon, Savlugu, and Nmion, which are all in the northern Ghana. The districts received approximately equal amounts of rainfall since they all fall within the Guinea Savannah agroecological zone. And then the soils are generally loose, shallow soils. And then in terms of fertility, they ranked with Tolon being the lowest and then the Mion side being the highest soil fertility. Now, in order to do the assessment of these impacts, we had to rely on an experiment that was carried out in 2014 in a similar environment. And then this experiment was used to calibrate and evaluate the absent model for further study. The calibration and evaluation after receiving appreciable results were now used for the long-term assessment of the sustainable intensification practices, relying on weather information from 1984 to 2024. The varieties that were considered were 2, an early maturing variety, extra early maturing variety, and then an intermediate variety. Now, we also used 3 levels of inorganic fertilizer, 030 and then 60, and then 3 levels of organic manure, which is 2005, and then 2,500 and 5 tons manure. Planting window ranged between 15th May and then ended on 30th July with 2 weeks intervals. These were based on information that was received from an earlier survey, socioeconomic survey that was carried out in the region. The assessment of the practices were based on 4 main criteria. 1, the yield change that was obtained from using the practice. 2, the internal standard deviation that was also obtained from the practice. Then the yield stability, and as well as the coefficient of variation. Now, in terms of the results, the effect of the fertility treatments were such that the combined application of inorganic fertilizer and then manure generally improved yields greatly across all the sites and across all planting windows. This is because of the improved effect that organic manure has on the soil by improving soil structure as well as enhancing nutrient cycling ability of the soil. This is the results from the ANOVA that was run for across the locations for grain yield. It was realized that the 30 kg N with 5 tons manure produced the best yields. These yields were significantly higher than the other treatments across all the 3 locations that were studied. This is the distribution of how the yields were across sites and then across 2 different, across the 3 sites for a particular sowing window. This is for the earlier sowing window. Also, when you look at it, you realize that the yields that were obtained from the intermediate variety were higher than the yields that were obtained from the extra early maturing cultivar. In terms of the yield responses by means of percentage, the response were highest at the Toulon site. This is a site that has inherent poor fertility without fertilizer applied. The yield response to fertilizer application of 30 kg was 153, whilst at the Toulon Mion site, it was 58%. This is because the Mion site already has a good amount of organic matter in the soil, which makes it more fertile, and hence when yields fertilizer is applied, the sort of change that is gotten in terms of yield is not that much. Now, doubling inorganic fertilizer across the sites also resulted in yields averaging 42% for the early maturing variety and then 53% for the intermediate variety. As I mentioned earlier, the 30 kg N recorded the best yields across all the treatments. Now, this is the effect of planting window. Earlier planting windows generally recorded the highest yields compared to the later sowing windows. This result is corroborated by results that had been obtained by another researcher within a similar environment in 2018, where she realized that early planting is generally associated with higher rainfall. Hence, the yields from early planting are better than late plantings. This is the results from looking at how when planting date changes, yields also change. You see from the results that from the Savulu and Mion side that when planting window is delayed, the yields decrease also increases. This is especially more prominent when no fertilizer is applied. Now, in terms of the matrix, that is internal standard deviation, yield stability, and then coefficient of variation. The Mion side had the highest stability. These are sites with the highest soil fertility. However, at the Toulon and then the Savulgu sites, there were no significant differences between them in terms of yield stability. In terms of internal standard deviation, no significant differences were observed across the sites. However, when we looked at the coefficient of variation, Toulon, which had the lowest fertility, had the highest variability in yield, making it more risky for maize cultivation, whilst the Mion site had the lowest variability coefficient of variation in terms of yield. Now, we looked at the metrics on location basis and realized that at the Mion site, the 0N and then the 0N with 2,500 were significantly lower than the treatments from the other fertility levels. Also, there were no significant differences for internal standard deviation. And then also the 0N with 0 manure significantly showed higher variation in yield compared to the other treatments. This was similar to the other 2 sites in the study. Now looking at the effect of planting date on the matrix, at the Mion site, there were no significant differences between the yield stability. However, in terms of the late planting, they were significantly higher than the early plantings. All right. And then similar results were obtained for the Toulon and then the Savulgu site. Right. Now, this is considering the effect of cultivar across the 3 locations. It was noticed that no significant differences were obtained from using the 2 different cultivar across the locations in terms of internal standard yield stability and then coefficient of variation. In conclusion, can be stated that combining modest inorganic fertilizer with manure, 5 tons of manure coupled with early planting generally resulted in the highest yields across the sites. Also, the intermediate variety yields better than the short duration varieties. Okay. These are the references that were used for the study. Thank you.
Thank you very much. You managed 20 slides. Great. So next speaker is Ms. Lotte Wojtjes. Or Wojtjes. Wojtjes. Okay. Your talk is about intercropping immature oil palms with food crops.
Which button should I press to go to the next? Just that one. Okay, thanks. There we go. Yeah. Good afternoon, everyone. Thanks for joining this session. So my name is Lotte Wojtjes. I'm an assistant professor from the Plant Production Systems group in Wageningen. I'm a bit of an oil palm lover, as you can also see on my shirt. Sometimes it's hard to admit, but I admit it here. So I would like to tell you something about work that we're doing on intercropping with food crops in immature oil palm plantations, focusing on Indonesia. So just to give you a brief background for those who don't know about oil palm. So in the beginning, those oil palms are, of course, not productive. And that leaves a lot of, they're also very small, right, when you plant them. So there's a lot of empty space between unproductive palms. And many smallholders in all parts of the world practice intercropping with food crops in those immature oil palm fields. Bigger plantations have discouraged this practice because they worry about resource competition, especially light between the palm and the intercrop. So there's actually not much scientific study on intercropping practices, although there is some early work from Benin, for example, of very good quality. So we participated in a project called the Sustain Palm Project, which actually ended this month, funded by the Dutch Ministry of Foreign Affairs, which focuses on 4 pillars to improve the productivity and circularity of oil palm systems in Indonesia. And one of the pillars is to integrate livestock and crops in oil palm. So that's where this work also fits in. So our research question was, what are the effects of intercropping with watermelon and banana, the 2 crops that we currently work on the vegetative growth of young oil palms? Of course, what we would love to study is yield, but that's why many people don't like to study perennials, because they take a very long time before they actually start producing yields. So in any short research project, you have to do with vegetative growth unless you work with mature crops already. So we did an experiment, but I have to admit it's not really an experiment. This was actually a company that was running production of watermelon and banana in an oil palm plantation. And so we basically laid on an experiment, but it's not laid out as in an experimental design, which is a pity. I will explain later what the effects are. So we work in a field in a company plantation, and the first year they intercrop with watermelon, and the second and third year they intercrop with banana. And then no fertilizers were applied on the immature palms. There were fertilizers applied on the intercrops. So in the second year, we also applied to most of the palms that we observed fertilizers. And the intercrops are managed by independent farmers who are brought into the plantation and who get the benefit from the production of the intercrops. So this is an overview of the experimental treatments. So we have a monoculture as a comparison, and then we have either banana after monoculture, or first watermelon, then banana, or monoculture, or watermelon first, and then no more intercrop. So those are the 4 options, the 4 combinations. And then we have a different number of plots for the different treatments, because this was not a well laid out experiment, but we just overlaid it. And most of those were fertilized, that's the F, and 1 or 2 plots for each were not fertilized. But what we made the observations on were the individual palms. So those are the unit of measurement. So these are you see here an overview of the numbers that we have. So what does it look like schematically and in the field? On the left side for you, you see the watermelon in 2 rows between the palm rows. And on the right side, you see the banana also in 2 rows. And you can already see why there would be a concern about maybe light competition when you see the banana already being taller than the watermelon. And this is what it looks like on a map. So you see that the watermelon are the light green and the yellow colors. They sort of cluster together. The banana is the brown and the yellow, and then the dark green are the monocultures. So again, as I mentioned, this is not a nicely randomized design. What did we do? In the oil palm, we measured the growth. And we also, by the way, measured in a number of smallholder fields, which were also practicing intercropping. So we did some additional measurements in smallholder fields in the same region. And we measured, among others, we counted the number of leaves, the size of the leaves, and the height of the palms. I must admit that the picture is not from this experiment, because in the smaller palms, the leaves are actually much smaller. I'm measuring a mature leaf, which is about 6 meters long. So these are the results. You see the treatment combinations at the x-axis, and on the y-axis, the different things that we measured. And the second and fourth double, what do you say, boxes are the where you have watermelon. And what you see is that the watermelon, the treatments where we had watermelon first, in general, do better in terms of front length and palm height. And we don't really see a clear effect of the banana. We also don't really see a clear effect of fertilizers. Again, we didn't do a statistical analysis because this is not a properly randomized design, but it's just to give a feeling for the kind of effects that we see. And this is the results from the smallholder fields, where we see I only present the front length here. On the y-axis, you see the month after planting. So this was just a one-time observation in smallholder fields with either banana, monoculture, or watermelon. And again, we don't really see a pattern of lower or higher or larger or smaller fronds, depending on the different treatments. So basically, from this sort of observational study, we don't really see any negative effects of the intercrops on the palm growth. And we might actually see a positive effect of the watermelon. In addition, the watermelons provide a yield of about 20 ton per hectare, very substantial, giving a revenue of roughly 4,000 euros. They also require a lot of inputs, by the way. And what we also found is that smallholders actually really appreciate this intercropping, because the owners are usually not the ones who also do the intercropping, so they invite other, maybe landless farmers in their communities to come in and do the intercropping. So this improves their management of the immature fields, which are otherwise usually left unmanaged. But it's also good for the social cohesion, which the smallholder specifically mentioned as an advantage. Now, a big downside of the experiment, which it really isn't, is that there is a confounding effect of landscape position, because the watermelons require irrigation, and so they need to be close to a water source, so they were lower in the landscape. It is quite likely, though, that the palms benefited from the fertilizers and the water that was applied to the bananas and the watermelons, especially because the experimental period coincided with a relatively dry period in Indonesia. But it's also likely there's some competition for light with the bananas. I say here that next year we will measure the intercropping effects on yield, but we already have some numbers, and it seems that actually the yields in the oil palms combined with banana are much actually higher than the yields in the monocrop, so that would be nice. But again, there is this landscape effect that is probably confounding. All in all, we really urgently need more long-term and really properly randomized experiments, which in oil palm always become quite big, because you need the plots are really large, but still it's really urgently required in order to be able to give good recommendations and suggestions on how to do this crop integration properly. With that, I would like to thank all the students who went to the field to collect data. I also want to thank the company, both the company that owns the plantation company, but also the company who arranges the intercropping, and the smallholders for answering all our endless questions and allowing us to take measurements. Thanks.
Thank you. Thank you for the very clear and concise and well in time presentation. We are gathering the questions, right? This is now the last presentation by Mr. Martin. And after this presentation, we will discuss all the 4 presentations together.
Thank you. Hello, everyone. My name is Martin Raphael Tendeng. I'm a PhD student in agriculture science at Gaston Berger University in Senegal. Today, I'm pleased to present to you one part of my research that was done together with Amadou Diallo. The title of my presentation is Performance of Lettuce under Different Shading Level in Saint-Louis, Senegal. Let me briefly outline my presentation. I will start with the introduction, then more on the material and method, followed by the result and discussion. And I will end with the conclusion. The Niayes region is coastal northern Senegal is the main horticultural area in Senegal, producing almost 80% of the country's fresh vegetable, mainly onion, tomato, sweet pepper, and lettuce. Vegetable production is the main source of income for more than 90% of households in Niayes. However, the vegetable production in that region is increasing, challenged by climate change, effect including increasing temperature, high solar radiation, and intense evapotranspiration. This reduces both the yield and the quality of the vegetable. A potential solution to this challenge could be the integration of vegetable into agroforestry system, for example, fruit trees. However, no data of the suitability of such system are enabled in Senegal. Our reason could be that study of agroforestry system are very complex and take a long time. Shade net could imitate shade of trees with comparable microclimate conditions. Thus, they could be tested as possible adaptation strategy and for accessing specific response to the prevailing microclimatic condition under shade. The objective of the study were first to assess the effect of different shading level on agronomical performance of lettuce, and second, to identify the optimal shade level to maximize lettuce yield. The experiment was conducted at the experimental farm of Gaston Berger University. The experimental design was a completely randomized block design with 3 replications. We used different shade net with 35%, 55%, and 75% shading in the trial. The control plot had 0% shading. Each plot had a size of 2 by 2 meters. For measurement, we used 6 plants randomly selected in the core of the plot. This has been done 8 weeks after planting. We access the 3 variables, namely first, total biomass, which includes the roots, second, above ground biomass, and third, marketable yield, which means that all our damaged outer leaves were removed. Data were then processed using analysis of variance and post hoc decay tests. I'm coming now to the result. This photo of 6 plants harvested from each of the plot clearly shows that the plants cultivated under high shading level performance better than those in light shade or in full sun. Our statistical analysis confirmed the first impression as shown in the next slide. Here are the results of total biomass of lettuce, including the roots. Shade level had a significant effect on total biomass. The 55% shading showed the highest biomass compared to control. Lettuce ground under the 55% shade had a mean of almost 150 grams total biomass, while those cultivated full suns had only about 70 grams. Similarly, shade net level had a significant effect of the above ground biomass of lettuce. Lettuce plants grow under the 55% shade net showed the best performance as compared to the control. Even more clear was the result for marketable yield shown in my next slide. Here again, we can see that the 55% shading showed the highest marketable yield compared to the control. Lettuce grow under the 35% and 75% shade nets showed medium performance. I'm coming now to my conclusion. First, the study showed that shading did influence the performance of lettuce with best performance under 55%. As compared to the control in full sun, intermediate shading could thus a good strategy for lettuce producers could probably provide similarly intermediate shading levels. Instead of using shade net vegetable, such a lettuce could be integrated into agroforestry system for enhanced crop productivity. Finally, we therefore recommended taxing lettuce production under trees. We are now planting test trials and expect that lettuce is a suitable species for integration into agroforestry system. With this, I am ending and would like to say thank you.
Thank you very much. Very good in time. So I guess you collected your questions, and maybe we start with the first presentation of the last 4 talks by Ms. Kiboi. Are there questions related to the long-term experiment in Kenya? Yes, Daniel?
Thank you for the presentation. A question related to the conclusion that there's no difference in the productivity between conventional and organic management systems as you studied in the field experiments. My question is related to the nitrogen inputs, which were in the high level about 220 kilogram nitrogen per hectare and year. How do you think it's possible to get these amounts of nitrogen on a larger scale? This was experimental fields. As far as I know, you used 2 types of inputs. Some of them, the Titonia leaves collected. How do you think it's possible to upscale this with the availability of the inputs?
Sorry, this with Ms. Kiboi was.
Okay.
Yes, thank you very much. So for the N inputs of 220, this we were considering the farmers, the large-scale farmers who are mainly doing this for commercial production. And how possible it is still a challenge. But what we do now is that we're working on a series of experiments to improve the compost quality to meet these N levels. So it's still ongoing on how possible and what feedstocks we may use to improve the compost quality in terms of N content.
Okay, there was a next question. Is it related to this presentation?
It is related to this presentation. So I'm just asking a question about the trial you conducted, because what I have seen is that you do it using a crop rotation. I don't know if you are planning or it is something that already been done using a continuous cropping system. Maize without having some crop rotation. I'm asking because what I have seen is that you conducted the trial using crop rotation system with maize and other vegetable crops. So I'm asking if in your treatment you have something like continuous cropping system instead of doing some crop rotation. Did you try to look at the effect on the continuous cropping system instead of doing crop rotation?
Do you mean continuous maize? Continuous maize?
Thank you very much. If you allow me, is that in Syscom, it is always with the rotation, but I have been privileged to work in other experiments. So it's maize throughout for 6 seasons, and the results were still similar to that. The inputs really did not influence the stability. Yeah. We have
Last question regarding Madina. This is the last question regarding the first presentation.
Thank you for your nice presentation. My name is Madina, and I was wondering, it's a bit difficult to get long-term data, experiment data set on crop rotation for modeling purpose. Are you planning to apply crop modeling on the data set you got? For example, for exploring more organic fertilizer treatment impact on your yield stability?
Sorry, I didn't get, am I planning to?
Use crop models.
To use crop models?
Modeling.
Modeling.
Reproduce your
Yes.
experimental data.
Yes, I agree with you. Data management, especially for long-term experiments, it's not easy. And this is something that we are still working on. So we have consolidated all this data. And soon we're going to do some modeling with the data in partnership with ETH Zurich. So currently, not yet done, but there is the plan. Yes.
Great. Thank you very much.
Thank you.
I think that's a good solution. May I ask Mr. Fredua to come to the microphone, and then you can respond to the questions related to your presentation. Are there questions related to the second presentation? Oh. Seems there are no questions. Yeah, there's one question.
Okay.
Over there. Sorry, we need a microphone for the audience in the Zoom.
Hi, thank you for your presentation.
Yeah.
I was wondering, a bit similar to the other question, you recommended a mixture of chemical fertilization and organic manure fertilizer, and you said the best would be the 30 kilogram N plus 5,000 kilogram manure. And again, it is on a small plot rather. Do you think this is really feasible for farmers in a larger scale to put 5 tons of manure to each hectare? Do you have so much manure in northern Ghana?
Okay, so actually, manure usage is being widely promoted now in the northern Ghana. So there are products such as Fertisol, which is being vigorously promoted by the agricultural extension agents. And then, fortunately for us, most of the farmers within the system are small holders. They are not large-scale farmers that would require a lot of manure. So the system works for us. There's enough manure if it needs to be applied. Yeah.
More questions? Okay, thank you.
Thank you.
Thank you, Bright. Next presenter, Ms. Wojciech. Are there questions related to the intercropping of oil palms with watermelons or bananas? There is one question up there. Lecture room is too big.
It's too big. Free fitness workout.
First of all, thank you for your presentation. How did you choose banana and watermelon? Is this an intercrop that you saw there in this area already, or was it just guessing?
Well, good question. Thanks. So we didn't actually choose it. So this is, there's a company which connects landless farmers with land, in this case, oil palm plantations, and they have agreements with suppliers. And in this case, there was a demand for watermelon and banana, and there were farmers able to grow those crops. So that's how it happened. And that's also the idea, to make it more scalable, is to work not just to grow anything, but really to work with suppliers who are interested in getting these products. I mean, there's many other crops you can intercrop with oil palm, most actually. So.
Another question there.
Thank you for your presentation. It was so interesting. I have two questions. First one, as far as I understood, you said the intercropping with watermelon had positive effect on the watermelon yield. So do you know any scientific reasons behind it?
Sorry, we didn't study the effect on the watermelon yields.
Only effects on oil palm.
Yeah, so we of course we monitored the watermelon yield, and I wouldn't know by heart what would be the watermelon yield in a monoculture. I also don't know exactly what the relative planting density was. That's not what we worked on.
Okay. Did you also study about the crop protection aspects, like any common pests or diseases?
I mean, crop protection was carried out according to a standard operating procedure in the watermelon and the banana. So a full crop protection carried out, that's pretty intensive. I don't know exactly which pests and diseases were present, but they were controlled for.
Thank you.
And control of diseases for the oil palms? Was this necessary?
No, I mean, oil palm in that sense is quite nice because it has relatively few problems with pests and diseases. And the key disease that is in Indonesia will come up usually when they're older.
Okay.
So there was no control.
Last question.
I was thinking about the watermelon again because you said it's a very water-intensive culture. Are you planning to continue this project? Because this might be interesting about the water retention from the palm tree roots and the water management in the watermelon. So this might also be an improvement.
I can't hear you entirely well.
I was just wondering maybe about the positive effects of the water retention of the palm tree roots on the water management in the watermelon, because you said watermelon is very water intensive.
Yeah, I would not expect any positive effects from the oil palms. I would expect competition, especially as they grow. So in this case, I would say that it was mostly the oil palms which benefited from the water from the watermelons, not the other way around. And at some point, I mean, the shade is a problem first. So at some point the canopy closes and basically you get too much competition for light and the watermelon wouldn't work. That's also a question if there's any shade tolerant crops that you could grow below mature palms, but the shade is really intense. So that's a challenge. Yeah.
Okay, thank you.
Thanks
for answering all the questions. And now we will discuss the last presentation by Mr. Tendeng about growing lettuce under different shading levels. Are there questions related to this last presentation? Daniel?
Thank you. Did you make some additional assessments which may help to explain the results? For example, the temperature or soil moisture levels at the different shading levels?
Yes. Yes, je peux parler en français, peux-tu traduire?
Voilà, je peux répondre moi.
Ok, donc
Can you
je réponds
translate
après.
in English?
Yeah. Effectivement, on a mesuré la température parce qu'on a posé au niveau de chaque shade net des petits matériels qui nous permettent de mesurer la température, l'humidité, et en même temps, on prenait les données d'humidité du sol.
So they did some They took more data, not only about the phenotypic variant observation, so they were able to have data on moisture content, temperature, but also other climatic data, something that was not maybe presented in this presentation.
Maybe I can add, so how do you explain the positive effect of 55% shading on the biomass of the lettuce? Is it, I mean, you reduce the incoming light by more than 50%, but you have a higher vegetative growth. How do you explain this positive effect?
Comment expliquer l'effet positif? Par exemple, de ce que tu as observé, 55% en fonction de la teneur en biomasse, par exemple. Qu'est-ce qui fait que tu as une teneur en biomasse beaucoup plus importante sur les 55%?
Bon, ce qu'il explique, c'est le microclimat du niveau d'ombrage de 55. Parce que lorsqu'on a fait la corrélation, on a vu qu'au niveau du microclimat, 55%, on a moins la température, elle est régulée, aussi l'humidité, elle est régulée. C'est là-bas qu'on a des conditions de développement de la laitue que les autres niveaux d'ombrage.
So it's just about the microclimate you create when you use the 55% shade net. So when you have it, they try to do some correlation between what they have and the temperature. So they was able to see that they reduce the temperature under the shade net of 55%, which give more microclimatic condition for the lettuce to grow under well condition.
Okay. More questions? Okay, then thank you very much, especially the presenters for presenting, the spontaneous translator. Okay. The next question is half an hour, right?
Yes.
We are well timed.
Yes, we finished within time. Yeah, we finished. Yeah, within exact time.
Perfect. Exactly.
Was all the presentation till now.
We have already the new.
Yeah,
That's the basis for the next session.
yes. I have to check the folders.
Okay, thank you for helping.
Thank you for it was really nice that I am here. And these are the interesting topics. Yeah.
So see you perhaps next semester.
Yeah, yes. Yeah, this semester I hope.
GIS?
Yes. Bye bye. Have a nice day. Wait a second. It's not a nice person.
Okay.
And now it's working. Okay.
Okay, thank you.
So, session yes. Is something like that? You upload it online?
You can
like this, but problem is technical to the internet. Doesn't work later, I don't know. You have this? Yeah, because I don't want to, if there is technical load there later, so it's oil setup here, so it's, you have laptop and another pen drive. Straight.
Yeah, but I don't. I have another one, but mine is still busy. The other thing we can do it, I think, email. If it's, but let me try this. I want also to check mine.
Sorry, I have a problem with my presentation. I want to copy the new version.
Sorry, you want to,
Copy the new version of the presentation.
yeah, you can give by the pen drive, you have?
Okay, no, I don't. I have it on my system. Do you have or can I send it to your email?
Actually,
Okay,
Same.
I can borrow.
I did not put it yet, but it's fine.
yeah. Okay, let me see.
That is. That one is long.
Yeah.
Is it the same?
It changes to, I don't know, I open it's still the same. Sharing and scope. Okay, I have to start again then later.
Okay. Oh my God, I left basketball.
Yeah. So we are online now.
Everything's fine? Come on y va?
Yeah.
Good luck. Good afternoon and welcome to the first, the second, or the first part of the crop biotic stress component. We had a lively poster session this morning, and this afternoon we continue with the oral presentations. And this is basically to, okay, not camouflage everything, but to just say, okay, the missing audience we compensate with excellence in science. So welcome, my name is Stefan Winter. I am a researcher at the Leibniz Institute, DSNZ, who somehow is affiliated to the Trubentag since a long time. And I'm guiding and chairing you through this session. Once again, as I said already this morning, we have an enormously broad spectrum of topics to speak about. And this is a strength of the Trubentag because it makes us sensible for the topics that we all deal with in our related discipline. And
Again, I'm enormously grateful that this year the Drobentag has invited a keynote speaker. So I would like to welcome, Dr. Fatia Kamis, who is currently the director of the Integrated Bioscience Platform of ECP in Kenya. As we had already a distinguished laureate from yesterday, who's also in our audience, I would like to give you the floor to enlighten us on molecular tools for sustainable management of pesticides.
Good afternoon. And today I will give you a topic on molecular tools to augment what was yesterday depicted by my Deputy Director General and also my supervisor on IPM systems. So to start with, I know yesterday he did an introduction, but I would like to also augment that in this audience, we have both our former DG and also our current DG in the audience. And thanks, Professor Bugmeister, for this invitation. And maybe to also mention, we have as ECP our footprints throughout Africa. We work within Africa all over, and of course, we have global partners. Now to go to the topic of the day, and also the theme of the Drobentag 2025 on planetary health. As we know that our population is growing at a faster rate, and especially in Africa. And this population growth is predicted to reach around 9.7 billion within 2050. And of course, we have to increase, the demand for food will increase drastically and is still increasing as we are now. Productivity has to increase, in fact, 60 to 70% to meet the food demand. And of course, out of this population that is increasing, we have urbanization that is taking place. It is predicted that out of 10 people, 7 people will move to cities. And of course, our dietary requirements are shifting. In fact, there are people who go from non-vegetarian to vegetarian. We have those shifting from vegans to feeding on animal protein and all this. So also now to catch up with this demand for food, we have to increase land for productivity, but yet we also have to, there's also land that is utilized for built environment to accommodate this increasing population. There's solid waste accumulation because of the population, first due to food waste, food loss, and our lands are degrading at a faster rate due to climate change and also overuse in agricultural or other anthropogenic activities. So the challenge posed by this are several, and one is food and nutritional insecurity. Of course, we are still grappling to meet the food, to meet this SDG 2 of zero hunger, but yet, since the population is increasing, about 20% of the population is facing hunger in Africa. We also have our annual food bill increasing threefold from 35 to around 110 billion. And vegetable consumption, around 90% of the countries are not meeting their per capita vegetable consumption. And we also have a lot of prevalence of hidden hunger, be it micronutrients or also anemia is prevalent. You could see this. So the second challenge, as much as we are to produce more, but we have challenge in fertilizer production, and we've seen the increasing demand of this. And recently, when we had conflicts in some areas, we could not get fertilizer to sustain our production, and the demand is still going high. Another big challenge is global demand for meat. We talked of dietary shifts. So there's a change in culinary habits where we are demanding more of the animal protein. And this is predicted to go higher by 2050. As this is going high, we also have to sustain the animals. So also the demand for the feed additive, the protein source, has to increase. And we have to do the trade-offs between some of this protein additive to human food or to animal feed. So the solution is sustainable food production systems, and agricultural transformation or sustainable agriculture is key to sustainable development. But this sector is yet to reach its potential. It is constrained by numerous biotic and abiotic factors from pre-harvest to post-harvest, which includes pests and diseases, poor soil management, poor management of this pest, and the climate change also is also adding to this. And post-harvest, we have the storage pests, we have infrastructural challenges, especially in Africa, poor storage and cold chain for this food to reach the markets, and also effect of. So what is the effect of all this? It's leading to poor yields, which cannot sustain the systems. Now to go to the biotic factors, ranking high among all the biotic factors is constrained by pests and diseases. And we have a cocktail of indigenous pests and diseases in Africa, which are wreaking havoc to our yields. But also now, due to poor or porous borders and regionalization and globalization of trade zones, transboundary tourism, and poor phytosanitary and infrastructure and capacity, we are also vulnerable to invasive pests and diseases, which are a threat to livelihood. And these pose two constraints. First, they reduce the yield, and secondly, they encourage overuse or misuse of pesticides. And this greatly impacts on the economic, social, and environmental fabric of our communities. So the solution for this is sustainable management of these pests and diseases using integrated pest, using the ecologically friendly and holistic integrated pest management system. And what are these systems? Normally, they consist of many approaches. We have from cultural practices until good agricultural practices, use of soft pesticide, use of biopesticides, and other habitat management methods. But the key component of any IPM system, we have several components of IPM system, from prevention and suppression to reduction or increase of yields and quality of product through reduction of pesticide use. But the key component or the key features is identification or rightful identification of either a pest or disease. And here we come in with molecular tools. We have a number of tools that are available in molecular biology, from simple identification using PCR to population genetics to genomics. And we have the different omics, from transcriptomics to genomics, mitogenomics, and also other molecular tools that can be used. And essentially, you can use these tools for pest identification. We can use them to evaluate resistant mechanism in pest population. And we can use these tools for host interaction studies and also population genetics. So the tools have several advantages. They're handy and advanced. You can see that each year we have these tools advancing more and more and being even user-friendly. They are not influenced by the environment. They are stably inherited. They are useful in several stages of insects. You can use it. It's unlike normal or taxonomic tools where you have to have intact organism for you to identify. And we have a lot of resources available online that you can use to do your identification. And of course, there's the declining cost of this. First, genomics used to be 3,000, but now it can go as low as $300 or euros. And then ease of use and application. So why the molecular tools enhance IPM application? First, accurate identification will ensure effective and targeted management approaches, so to save time and resources, effective surveillance and monitoring. So these tools can ensure that you get your rightful identification. And of course, you can put timely intervention of these management options. And the tools also, you can track the pest and disease epidemiology and also track the invasion histories. So where you can get your biological control agents nicely put in place. Then we can also track resistance so that we can cap this resistance and ensure that we sustainable management. And the tools also offer advanced management options like use of RNAi or CRISPR gene drives, marker-assisted selection, and so on. So I'll give you a case to augment what was yesterday given by my supervisor here. Of course, in Africa, I'll give you the case of fruit fly invasion and how we used molecular tools for this. Of course, in Africa, we have a cocktail of native fruit fly species from the Dakas, from Ceratitis, and also from Bactrocera genus that have neutralized to the African environment. But of course, we also have a lot of invasive fruit flies. And currently, we have a looming threat by Bactrocera zonata, and also we have already Drosophila suzuki in Africa. So with this species, which was detected in 2003 at the Kenyan coast, by 2005, this species was described to be Bactrocera invadens, a very invasive species that found its way from the easternmost point of Africa to the western point in just a few years, within a few years. And it had a wide host range, very highly invasive, very highly destructive, and it reduced yields of just mango crop from 40 to 80 and to even 100%. But importantly, also it encouraged overuse of pesticides. And with this also, we were hit by loss of exports, ban of trade for certain fruits to lucrative markets that we used to have. And of course, we lose revenue. All this culminates to socio-ecological implications for African countries. But the beauty of the pest or the downfall of it, it had an identity crisis. It was such a headache for the taxonomist. First, within the pest population, wherever you pick it, it exists in different morphotypes that were thought, we were not sure, is it a species, just a population of the species, or is it a species complex? So first we started with simple tools. We constructed a microsatellite library to unravel the identity of this pest or the population genetic structure. Where do we end with bee invaders and where do we start with another species? But actually when we did our first use of molecular tools, we inferred very low variability between these populations and within also a population. So at least this was a confirmation that we are dealing with one species as not a species complex. So that was one. Then we went further to do just morphometry and see if all these morphotypes and populations within Africa, are they just a homogeneous population? But actually with these tools, we also included some closely related species. And we opened a Pandora box because the pest with other closely related species could not be separated using this tool. So again, we went to employ molecular tools and to find out, is this Bactrocera invadens the same, one and the same to Bactrocera dorsalis? So when we did our molecular tools, also everything, there was no genetic variability between B invadens and B dorsalis. So as we were told yesterday, of course, IAEA came in and brought a new CRP, Coordinated Research Program for the pest. And using integrative taxonomy, using different tools, and of course, the center point was molecular biology, we could synonymize these two pests. And the implication of this is easing trade barriers between countries. At least some of the produce from Africa could be exported. And some of the pre and post-harvest measures that had already been put in place for this old pest could be used in Africa to do for management of the pest. And of course, it isn't also bureaucracy between countries, especially when it comes to material transfer for biological control. I'll now go to a case where we also highly use molecular biology tools, and this is the case of tomato. Tomato crop is a socioeconomic crop which finds its way in Africa. It offers employment for women and youth, and also it finds its way in every table globally. And its production is so high, around 180 metric tons annually. But traditionally, this crop has been marred by several pests, from red spider mites to African bollworm to Rhizomyza leaf miners, white flies, and thrips. But in 2006, we had a pest that is of South American origin that hit outside its range to Spain in 2006, where it found its way to Africa. So from North Africa to Sub-Saharan Africa. And in 2014, we detected it for the first time in Kenya. And the pest was devastating. It encouraged overuse of pesticide and caused 100% yield losses for growers, which led to skyrocketing prices of tomatoes from 10-folds higher than what we used to buy, from 10 shillings to 100 shillings per kg. So using molecular tools, we did a local barcode library for this pest, where we were doing identification of the pest for other stakeholders around Africa, where each country, when they detect, they used to send us samples and we do the ID. But then to do biological control, we had to go back to our ecosystems and see which are other related Gelechides that are in the ecosystem and try to get biological control agents that can be used or can be used close hand in hand, and also offer maybe to also, when you are doing your dossiers for submission for any biological control agent, you have to know of non-target effects. So in the ecosystem, we got a lot of Gelechides in the ecosystem, which we picked and did a close variability studies using molecular tools. And actually, we found out that Tuta absoluta was closely related to Thoromia opaculella, the potato tuber moth. And before even we went further, because we wanted to do what is the relationship between the two. So we used further genomic tools like mitogenomics. And when we did this, the pest already was being described as Thoromia from Tuta absoluta to Thoromia absoluta. So this, our work also confirmed this, confirmed the close relationship that they share. And we also did niche overlaps using our knowledge of the mitogenome into modeling to find where these pests are likely to spread. So as I told you, the pests attracted a lot of pesticide. In fact, our first stakeholder meeting in Kenya, it was, we came to learn that the growers even were using acaricides to to spray to the tomatoes to save their produce. And when we did our evaluation of pesticides from different areas, from farm gates to the farms and also to the supermarkets, we got to learn that all the produce had a lot of laced with a lot of pesticides. And elsewhere, especially in South America, the pest is known to have developed resistance. So now, if we are going to still use a lot of pesticides, we have to make a move and see what pesticide can really work in our ecosystem and what we should develop for farmers to use instead of the insecticide that they are using. And actually, when we did our work, genotyping and also transcriptomes, we found a lot of populations that have developed or expressing resistant genes. And also most of the field population already had resistant genes in this population. And when we did further microbiomes, we could get a lot of bacteria that are associated with the resistant population being in abundance. And those that are useful to monitor or to suppress the pest, like Wolbachia, was suppressed. And when we went further to do modeling of the resistant gene, we predicted an exponential increase in insecticide-resistant genes in 20 years to come. So we also did immunocompetence using molecular tools because we as ICP develop a lot of biopesticide for different kind of pest. So with this kind of resistance that has developed for pesticides, we thought of doing immunocompetence of this pest against the entomopathogen. And actually, when we use entomopathogens, actually it reduces the competency of the pest. So all these have implication, and it culminates to what we develop as IPM system for different organisms. So it helped us to develop a comprehensive, ecological friendly management toolbox for the pest, and also safeguard against resistant development, and also advise policymakers on resistant development, especially with those specified pesticides that have been specified for our country. So I'll give you just a snapshot of whiteflies population in Kenya. It was traditionally known that we had Bemesia tabaci as the whitefly species in Kenya. In fact, all the management was geared towards Bemesia tabaci. But when we did our survey and using molecular tools, we found out that the most abundant population of whitefly that we had was Triloroid vavopararium, which is the greenhouse whitefly. And with this known fact, we started developing our IPM tools based on entomopathogenic fungi and also endophytic fungi. So when we had the fall armyworm, I know now we have a looming threat to Europe. So when we had this detection in Kenya, we wanted to do also the same, to follow our routine of doing a toolbox or a management toolbox or an IPM toolbox for this pest. And actually, when we were doing our research, we found out that there are different haplotypes within our ecosystem, and the abundant strain was the R strain rather than the cone strain, because we thought we will get the cone strain being on higher abundance. And of course, some of these species or the strain of this pest react or respond differently to semiochemical, to pheromones that have been employed. So it was good to start with a point of knowing which populations we had before you put in place management options. And like our normal, we put a management tool based on proper identification of this pest. So I'll give you also a snapshot of where we employed a lot of molecular tools. It was in citrus IPM. Traditionally, our citrus production, especially the highland citrus production, has been really constrained by an African psyllid the African citrus trioside, Triosa erythrae. So this carries the African disease, which is transmitted, the causative agent is Candidatus liberibacter africanus, and it causes the African greening. But in 2017, we had the detection of an Asian psyllid in Africa and in Kenya for the first time in 2017, and it was reported. This pest carries or vectors the lethal Candidatus liberibacter asiaticus, which is a lowland disease causing what is known as Hualongbing disease. So what we did is a delimiting survey to see the extent of spread of Diaphorina citri, and what we have, because it's been years since a delimiting survey was done for also the African citrus trioside. So when we did our work and using molecular tools, we could identify some of this. Like in Ethiopia, we got to know that we had both the insects, the Diaphorina citri, and also Triosa erythrae. And in Kenya also we have the two, while in Uganda we have the one. We had only Triosa erythrae. Then we went further to see, okay, what are they carrying in what are they vectoring? So in Kenya, in Ethiopia, we had both diseases, the Asian disease and also the African disease. And in Kenya, likewise, it has spread. Also, we had the African disease and also the Asian disease. And luckily in Uganda, we only had the African disease. And one of the interesting components that we found out is the Triosa erythrae, which is a highland pest, could vector the lowland bacteria, which I think it was a turning point to management that can be put in place. So to confirm where Diaphorina citri actually, to confirm its population, genetic structure, diversity, and also invasion history, we also use molecular tools. And we use both mitogenomics and also molecular microsatellite markers. And actually, interestingly, we found like there are two different invasion points into Africa. Like the Ethiopian samples were linking to US samples, while Kenya and Tanzanian samples were linking to China. And of course, this we confirmed using microbiomes. The same population of microbiomes for the two haplotypes, they were linking as is. The Ethiopian microbiome was linking to US, while the Kenyan and Tanzania were linking to China. And this has a lot of implication to biological control because these are our starting point to search for biological control agents. And of course, like our norm in ISIPE, we are still developing ecologically friendly methods based on our identity and the different populations that we have and subject to their responses. So the future of molecular biology, of course, first, we need a lot of resource mobilization, partnerships, so that we don't replicate each other, linkages and networking for us to know of new tools that we can use, and coupled to a lot of capacity building to produce cadre of future scientists that can do this work. And molecular biology can be used for field-based rapid diagnostics, second-generation management, food safety, breeding programs where to strengthen our, be it plants or insects that are based for food or feed, and also screening for effective management or resistance curbing. So all this fast identification and management to help have a strong IPM system, ensure food security and nutritional security, and also food safety, reduction in pesticide use, and improved or resilient organism, be it plants or insects, and do a lot of capacity building for sustainable food systems. I would like to acknowledge, because these pieces of work were funded by different donors, I would like to acknowledge our ECP management, staff and students that worked on different components, national and international partners that we worked with, and the ministries of agriculture. Thanks also to Professor Christian Bodmeister for the invitation, and also the entire TropIntag organizing team for the nice conference. And I would also like to thank our co-donors. Thank you very much.
Thank you very much, Dr. Thomas, for this inspiring introduction to molecular biology for insect control and management. I would like to allow a couple of questions, just not to blur discussion. Are there any questions? I have the first question and you the next. So for taxonomy, you use metagenomics. Is this high resolution needed for the general taxonomy of insects?
Not really. We first start with simple molecular tools before we do the full either in fact, we don't even go to genomics itself. We go to mitogenomics.
Yes.
Yeah.
So, but the mitogenomics, you find the most useful resolution?
Yes, because here we have a lot of like 12 coding regions. So it's easy to use any of these for identification. Yeah.
Thank you. Professor? Bürgermeister? Oh.
Thank you very much. My name is Maria. I'm from the Czech University of Life Sciences. My question was, what is the usefulness of using gut microbiome analysis for the clustering of the populations? Why do you use the gut microbiome?
Okay.
And not the actual genome of the organism? Thank you.
So first we do the full microbiome for two things, because here within the gut, you can get endosymbionts that we can also use for management. But at the same time, also we can get the pathogenic microbiome within the gut. So all this later on help us in management. Because if you have the pathogenic, you know how to handle it. If you have the endosymbionts, you can use for management. And also now that is an added function, because we could link that microbiome to the microbiome of a pest that is an older pest. Like now the US population is an older population. So if you could link to this, then it shows you the invasion patterns. Yeah. Yes. Yeah.
Thank you for a nice presentation. You mentioned the use of entomopathogenic fungi and bacteria in your biocontrol gadgets. What informs the inclusion of your entomopathogenic tools? Have you tried nematodes? What informs the inclusion of a component in your biological control?
Yeah, that's a good question. Actually, in ECP, we house a germplasm with several species of entomopathogens, from fungi to nematodes, and also we have several viruses within our germplasm. So when we have pest detection, and of course we want to do a quick tool, we go back to our germplasm and screen these insects against the germplasm. Whatever works is what we put forward. In fact, also we have bacteria, Bacillus thuringiensis, BT isolates. So we test all this, but we pick, because of course that organism will have been identified. So we go back and look at the germplasm, which are the closely related species that have worked against any of these isolates that we have. So that is our starting point. So we have also the nematodes, we have viruses, we have BT, yeah.
Professor Bürgermeister, and then the last question from Sam.
Very nice presentation, Fatia. Thank you very much. Just one question, RNAi, how do you see the prospects for use in Africa?
In fact, that will be so good, because we have a lot of insects and we have a lot of resistance to pesticides. This will be an ideal tool if we can do the silencing within our labs and proceed with it. Initially, what was not clear was the legislation, because it was not allowed. But in the recent past, this has been allowed in certain labs, and you have to be evaluated and you can do it. Yeah, so it is actually the way to go. And in fact, yes, you can even have it induced like a foliar application with the RNA sequences that you cut. So it can be an easier management method and a safer one.
Thank you so much for this wonderful presentation. I'm interested in your whitefly. So the whitefly Bemisia tapaki that you see that's not there now anymore in the Kenyan region. Did you try to detect virus inside the whitefly? Because we know cassava brown streak disease is a major concern for Africa, and whitefly is one of the vectors. So I'm interested to see if you try to detect any of the viruses in the whitefly, or if you still have the data to maybe try to detect later. Thank you.
Yeah. Thank you so much for that question. If you look at our work, we concentrated on tomato and vegetables agroecosystems. But of course, when we go to cassava, you get still you get the Bemisia tabaci, but also you don't get it alone, because in my experience with identification, we get also what is called Bemisia affair. We detected that. So this is quite complicated because, okay, we've not gone deep into cassava, but it will be interesting to find out what we have there.
Indeed. Thank you very much. I have to summarize and
thank you.
Thank you once again for this. Showcasing the excellent part, and I would like to invite Mr. Ibrahim to enlighten on test this simulation. And a benign warning, you don't have the luxury of our keynote lecturer to extend the time.
Okay.
So does this end, Genzi?
Okay. Good evening, all. Thank you for attending our talk. I'll be talking about the simulation and mapping of pest risks in maize in Nigeria. My name is Esther Shupel Ibrahim. This is a collaborative effort between my organization in Nigeria, my university, and my host institute, ZALF, here in Germany. So to give a background about Nigeria and why this pest is very important. Like Dr. Fatia said, Africa is growing, but Nigeria even growing faster. Currently, we have over 230 million people in Nigeria. About 21% of this population is unfortunately food insecure. Despite the fact that 70% of the population engage in one form of agriculture or the other, Nigeria is currently the 6th most populous country in the world, and the projection is that by 2050, Nigeria will be the 3rd most populous country in the world. So Nigeria has an area of over 900 kilometers square. 74% of it is arable land. Within this arable land, we produce a lot of cowpea, cassava, maize, peanuts. Nigeria is currently about the 13th largest producer of maize in the world, but the 2nd largest in Africa after South Africa. So having spoken about the potentials that we have in Nigeria, how much food we produce, this is a preview of the challenges that we have in Nigeria. We have issues with flooding, severe droughts, heat stress, but unfortunately even conflicts, especially from Boko Haram in the northeastern part of Nigeria. But most catastrophic recently is the issue of diseases and pests. For the sake of this study, I'll focus on pests. For pests, in maize, we have quite a variety of pests in Nigeria, but I'll talk about 2 major ones, which is the fall armyworm and the white grub. So this is threatening maize production, particularly in Nigeria. So I'll first talk about the white grub. It looks, it fits on the root of maize. So usually you see stunted maize growth, like I cycled, where you see very stunted maize looking reddish. When you dig underneath, you find this white grub feeding on the root of the maize. So for the rest of the cropping season, this is what happens. Where white grub fits on it, the maize is not able to grow further because if there's no root, it cannot take up chemicals. It can also not take up water. Now, in terms of fall armyworm, is feeding on above ground biomass. It means it feeds on everything above ground. So it feeds on the leaves, on the stems, on the on the grain itself, and even post storage. Now I give a short background about fall armyworm. It is a pest of the Americas, started in South America, migrated to North America, but also migrated to Africa in the year 2016, first reported in Nigeria. Now it is in all countries in Africa except for Morocco. I think the top three ones in Africa, Algeria, and I'll talk about why they are not found in these regions. Now, as the name implies, they move like armies. They move, so this is a depiction of how they look like. So they move like an army. It is very catastrophic because of how they move in colonies, but also because of their ability to breed. They breed very fast. One can lay up to 500 or 2,000 eggs. And then they can walk. So in one lifetime, they can move up to 200 kilometers. It means one can move from here to Brussels or somewhere like that. So its ability to breathe, its ability to walk, and the adults fly, that makes it very migratory. So when they arrive or on their path, they feed, they move. So this is just a depiction of what they can cause, get to your farm and then start to feed. In severe cases, empty out the farm. So with this problem comes also, just like Dr. Fatia said, issue of pesticides. So when farmers are faced with these challenges, what they do is keep on buying pesticides, trying to escalate the problem. Now over $400 million invested in pesticides. But then we know that causes environmental problems, causes a lot of health issues, deaths in Nigeria in 2020, over 270 people died just because of pesticides consumption. So with this kind of challenges comes what we can do as scientists. How do we approach the problem to reduce the issues of pesticides, but also smart applications of pesticides? That motivated this research. We had three main objectives. Now, there are a lot of research on the estimated yield loss when a farm is infested by pests. So mostly by surveys, but also by lab measurements. But then spatially, we don't have a knowledge of how much yield loss can be attributed to these pests, especially the ones that I was studying. So the first objective was to estimate yield loss as a result of either white grub or fall armyworm. Well, the second objective is that because farmers are trying to see how they can adapt, besides application of pesticides, they are shifting the growing calendar to try to avoid the period where these pests occur. So we wanted to test the shifting of calendar to see if it works in time to reduce the yield loss. But lastly, we're trying to also create an early warning system that the farmers can use for managing pests in maize production. So this is what I measured on the ground in Nigeria. So where you have healthy versus where it was infected by or infested by pests. So you can clearly see the yield is different. So how do we mimic that in a spatial model? So we use a model called an agroecological model called MONICA. It's a model for nitrogen and carbon. So it simulates growth of crops from sowing to harvest. So I'm trying to now, okay, how can I mimic that behavior and model it in an agroecological model to see how we can estimate the impact of pests? So we were able to achieve that based on the categorization of the pests. White grub being a root feeder, so we implemented a module for how maize reacts at different stages of growth, but if it infects different organs. Like for white grub, we implemented it just for the root because it feeds on the root. So in different stages of growth, what happens to the crop or how much yield is lost based on either white grub or fall armyworm, which is above biomass feeder. Now, the second objective, we're trying to see the shifting of sowing calendar. So Nigeria falls within 7 agroecological zones. So we're trying to see in every ecological zone, what is the growing calendar? So we adopted the FAO growing sowing dates, but implemented that in Monica to see what happens if you change the calendar. Now, the last objective relating to predicting the risks of fall armyworm. So in literature, we know that 2 factors are important for breeding fall armyworm, which is soil moisture and temperature. So this explains explicitly the distribution of fall armyworm globally. You can see that they are not found in Europe and other parts of the world because they are it's either too cold or too warm or too dry or too wet. That's why they don't appear there. So knowing that, with that knowledge from literature, we try to simulate the growth index, which is the suitability for it to breed, but also the stress index, what kills it. So we generated maps. We generated a module, a Python module incorporated in Monica to predict what happens from now until 2100 using CMIP6 data. So this is the first result, which is attributed to the first objective. We want to see how much yield loss spatially can be attributed to fall armyworm, but also to white grub. So we are estimating yield loss based on these 2 pests. For white grub, you can see that spatially it changes from different region to the other, but because we implemented shifting sowing dates, whether early, mid-sowing or late sowing, you can also see that it varies based on the infection, but also based on the time that it was planted. But also the beauty of this is shows spatially across Nigeria what happens when you have severe infestation or what happens when you have different dates of sowing. So in terms of the economic value, we have estimated that based on these 2 pests, we can lose up to 72 or over $670 per hectare based on the degree of infestation. So in a soft validation, we compare what is known from laboratory studies and also field studies. It collaborates because they report 5 to 75% yield loss as a result of fall armyworm. So in conclusion, based on this result, we say based on the yield loss can be different based on locations, but also based on the timing of sowing. In the second objective, like I said, we simulated from 2100, from 2021 to 2100. What is the growth index? You can see that in red is where it's severely or extreme risks, but also where we have no risks in white. But these maps are produced for every week from 2021. So we have an archive of it from 2021 to 2100, every week what happens. So it means that if a farmer knows his location, we can upload these maps and they can see what happens every week until then. So not only did we produce the growth index, you remember we produced the stress index. What happens? Do we have feasibility of this pest disappearing based on the cold index, the dry index, heat index, and the wet index? So in terms of this knowledge, like I said, this information is geotagged, it's raster-based. We built a simple early warning system, which is the last objective. So it tells in 7 days, every 7 days, help the farmer to know whether they're applying pesticides or not based on the suitability of soil moisture, but also temperature. So if these conditions are not met, it means the farmer should not apply pesticides, because currently in Nigeria, they just apply every week out of panic. So with this kind of information, they can avoid applying pesticides if the environmental and the climate conditions does not meet the threshold. So this implies information that is useful to the research community, but also the national and food planning of Nigeria and other similar countries, but also farmers and extension workers. If you're interested in finding out more about the findings of this paper, you can look it up here using the QR code. We've done this research, what is next? So we don't want it to remain within the context of a published article. We want it to be implemented to the farmers using SMS alerts or websites or public broadcasts from radio or TV announcements or cooperative or association of farmers, so that the farmers can get this information and potentially reduce the application of pesticides. But also we can use even smarter tools for precision agriculture like Internet of Things. Thank you for listening. I'm in the phase where we're trying to incorporate this from here to farmers. If you're interested in this, you can contact me. I'm open for collaboration. Thank you very much.
Thank you very much. We have one, two pressing questions. Christian, please.
Thank you very much. Very interesting. I have a question regarding fall armyworm. Let me be very frank. The way you sound, this sounds like we are back to 2017, 2018 when fall armyworm came, and you had these catastrophic losses, particularly in maize. I think we've moved on in Africa quite a lot. So what I've heard, what I've seen in the published literature, what I've heard from colleagues in the fields is that the loss levels due to fall armyworm has gone down tremendously. And people attribute this as increasing level of impact of natural enemies and concurrently much less frequent insecticide applications. Prior to fall armyworm, insecticides were hardly used in smallholder maize production in Africa. So this was really a novum. So I'm wondering whether this kind of modeling, which is kind of, I think, grounded in these catastrophic events of the first 1 or 2 or 3 years after introduction, whether this really still reflects the reality of maize farmers in Africa and the threats that they are exposed to. Thank you.
Okay. Thank you for that question and remark. Like you said, yeah, it's subsiding from what we had, both pests and diseases. That's because a lot of resistant species have been introduced. But based on our model, we're not incorporating this kind of scenarios. What we're looking at, because it is closely tied to climate change. So if you have a year where the weather conditions don't meet, of course, they will reduce, but that doesn't mean that the danger is over. If the climate conditions and the environmental conditions are suitable, they will rebreed, or if they grow resistant, and if the environmental conditions meet, they would breed. So that's what we are trying to see. Okay, we have data, climate data up until then. See every year, does these conditions meet or not? Is there likelihood of them to occur or not? So yeah, year to year, the impact changes, whether there's enough rainfall or there's too little rainfall or there's high temperature or low temperature, they interact with the environment. But with the agroecological model, it sees everything that happens every day and based on these climate conditions. So it looks at that before it predicts. And if you remember, within 2021, in my maps, I showed to 2040, there's high feasibility of them to reoccur. But from then onward, because the temperature becomes hotter or because the rainfall becomes more intense, the likelihood of them occurring are much minimal. But if you don't have this kind of simulations, there's no tool for planning. So those are the essence of this kind of research.
We have to stop this highly interesting, conflicting discussion, which is ongoing. I would like to now invite Mrs. Hasnim to the floor to speak about invasive species.
We have to take care of our planet. If you agree with me, let us try to stop this, human and animals. Now, I would like to ask you a question. What causes the highest number of deaths in this planet? The answer is not war, snake, or even drugs. The answer is a mosquito. Yes, it might seem impossible that something too small could kill all these number of human and animals, but it's true. According to WHO, mosquito bite could kill more than 1 million child every year. Mosquito transmit diseases like dengue fever, yellow fever, and Zika virus. Hey, wait, add an insect. Is mosquito only suck blood? Definitely no. Mosquito has other niche in ecosystem. They pollinate flowers. Male mosquito never bite. Female mosquito only suck blood to produce egg. So the normal niche for mosquito is nectar from plants. In our study, we focus more in the nectars. Another question, why mosquito bites someone instead of the other one? So we have like more than 200 attractant in our body attract mosquito. But the process why mosquito choose other person to another is not that clear yet. So we need to investigate more. The same thing is happening in plants. Why mosquito choose one plant to another is the same scenario. In this study, we will dive more in plant feeding and why mosquito choose one plant. Our main objective is to access the abundance and diversity of mosquito vectors across different villages in Baringo County in Kenya. So the study is in Baringo County, where we can find a lot of cows and goats. And the main economic product in Baringo County is honey. And they planted a lot of papaya, and also the area is infected with prosopis. So it's really highly invasive in the area. We start our study with consent form from the farmers because we will use a farm to set our traps. And then we use 3 types of traps: BG trap for day active mosquito, and CDC trap for night active mosquito, and OB trap for mosquito where wanted to lay egg, and or gravid mosquito. So here how we design our trapping technique. So we have like 2 CDC trap in each location, and in between we have 100 meters, and so in total we have 8 traps from CDC and BG trap, the day and night active mosquito. And for the OB trap or gravity trap, we have 4, and this usually in between the other traps, so it's like 50 meters away. So after we collect our mosquito, so video. Okay, it's how the mosquito look like inside the trap. And after that, we start sorting our mosquito. So usually when we come with the trap, we have another insect. So we sorting the mosquito, and after that, we place them in liquid nitrogen for viral analysis. And then we take our mosquito to the lab, and we start differentiate between the different species. So we will do morphological identification. And after that, we have 2 lines. We have plant meal analysis, and this is what we will focus here. And also we will do plant meal analysis. For plant meal analysis, we start first with a sugar test for the mosquito, if the already mosquito have the plant or not. And that is called arson sugar test. And when we got a positive mosquito from arson sugar test, we will do barcoding to detect if mosquito have like invasive plant or indigenous plant. And the same thing, it going for the blood meal analysis. So we will detect if mosquito have a blood from human or animals. And here, some of our findings. So we will notice that usually we have more mosquito in rainy season. So if you notice that here, we have like in rainy season, the mosquito in areas where they have the invasive plant and the areas where they don't have the invasive plant at almost equal number. In dry season, the area they have invasive plant, they have more mosquitoes. And even the highest number of mosquito in this study we collected in dry season, in where the invasive plant in the area. And what we did after that, we analyzed the mosquito and we detect if the mosquito eat already the invasive plant or not. And we found that. Finally, I just would like to say that we have to balance our ecosystem, because if we have more invasive plant, that means we have more mosquito. And if we have more mosquito, that means we have more diseases. So if we control invasive plant, we can control mosquito-borne diseases. Thank you.
Thank you very much. This is excellent. Because you also have bridged the gap between sort of like global health, invasive plants, pests and diseases, and human health. Is there any question to it? Well, I mean, so Platanium is an invasive plant, and I have seen this in the field myself. Once after it was invaded, essentially the whole field was covered in Platanium. So to compare Platanium with acacia is a bit of a weak thing. So if I would say something invasive plant, just call it Platanium, because I'm also fully okay in light of the dangers that this plant is. Any more questions? Very clear talk. Thank you very much.
Thank you.
From Nigeria to speak again about Spodoptera.
Right, thank you. Another talk on fall armyworm, another talk about Nigeria. So it's either fall armyworm likes Nigeria or... But I'm focusing on solutions. While we keep talking about the problems, I'm looking at the future. I've moved on. And one of the solutions here is entomopathogenic nematodes. It's a mouthful, and I'm researching what are the characteristics of this possible biological control agent that can be used against fall armyworm. And I looked at it using two models that I would like to call the triple IJ model and the shift model. I will talk more about this, but you might be wondering, entomopathogenic nematodes is a mouthful. Don't worry. These are tiny organisms. They are microscopic roundworms that infect and kill insects collaboratively with a symbiotic bacteria. They are environmentally safe, and they can persist in the soil and act as natural enemies of target pests. And the reason why they are this way, because they can be mass produced, highly reproductive, and can locate insects even where chemical spray cannot reach them. We've had several problems with insect pests. Fall armyworm was just one focus, and the solution or the promise is this tiny worm. I don't want to dwell so much about the biology of fall armyworm. Esther has done a good job, but if you want to read more, there's a very nice review article that took about 52 authors from 27 institutions to write, published recently, and they did a very nice job on that, so I don't need to dwell so much on this. But what interests me is that as the temperature rises, the climate changes, fall armyworm poses more risk. I wasn't trying to steal anything here, so scouting for fall armyworm. So the purpose of my research was to evaluate how does temperature affect the infectivity and productivity, and also the foraging ability, and also the resilience of these nematodes to physiological stresses such as desiccation, oxidative stress, or lack of oxygen. And the aims are to assess the influence of temperature on their virulence, on fall armyworm in this case, investigate how they forage and find their host, and of course, measure their tolerance to stress factors. And now to this models that I talked about, the triple IJ, what I did here was to look at all the data that I can collect and bulk them into models that can easily be applied for other research purposes. And one model deals with the impact of temperature on the infectivity or reproductivity, while the other model looks at the impact of foraging, their foraging ability and their resilience to oxidative stress. So taking on the first model, well, these are the meaning of the acronyms, triple IG and SHIFT. But taking on the first model, what we did was to have this insects in size boxes and introduce the fall armyworm under different range of temperature from 15 to 35 degrees Celsius, and then monitor the mortality of the larva. We used the third stage larva. We made sure that these are of the same size to control for any variability. And for the second bioassay, the reproductive ability of the cadaver and how much they can reproduce, we got the cadaver, placed them in a white trap, which is a designed Petri dish and a white collecting piece to get the nematodes out, and we count how much are reproduced. For the sake of time, I'm going to focus on one of the variables, which is the mortality of the fall armyworm. And after 10 minutes, we had a very poor mortality, which is expected. These are tropical isolates. And as the temperature increased, as expected as well, the mortality of the larvae also increased. It's not working. This is frozen. But then it peaked at 25 degrees Celsius. I guess there's a freeze here. Okay. It peaked at 25 degrees Celsius, and at 35, we noticed that there's a decrease in the mortality. There's a decrease in the mortality. So, I mean, there's a freeze here. I don't know why. Oof. Sometimes technology can be against us.
So heavy.
Can I escape and start it over again? Well, the end of the previous slide is that the mortality peaked at 25 degrees Celsius, but there was a little drag reduction in the mortality after 35 degrees. And the explanation to this is that at 25 degrees, either two things happened. The fall armyworm performed better. That was the optimum temperature, the isolate from that region, or the fall armyworm as Esther presented in her work, as we saw, that they could not really perform very well at that temperature. But at a higher temperature, the fall armyworm were also a bit more active as well. But the important thing I'd like to show on this slide is that with this model, with the type of data to collect, it can easily tell us how to integrate the infectivity and reproductive capacity using one conceptual tool. And also it highlights the importance of temperature as an ultimate gatekeeper for the use of biological control agents broadly, but entomopathogenic nematodes specifically here. The next experiment is on the foraging activity. Using the shift model, here I look at the stress factor and how the insects forage to find their hosts. And for the sake of this presentation, I will focus on the foraging ability. So for this experiment, we exposed the fall armyworm and the IJs in 4 substrates. First, a dry soil, a soil with moisture of 10% moisture content, 20%, which is a wet soil, and then on the surface of the maize leaf. And then we find out after 30 minutes of exposure, how many of these nematodes were attached on the larva. The second bioassay is how far can they go deep in the soil to find their hosts. So we have an array of nematodes on the surface, 10 centimeter down, 20, 5 centimeter, and then 10 centimeter, and then find out after 3 days, how far did this nematode go in search of their hosts. And on the dry soil, there were just a few attached on the surface of the nematodes. And at 10 centimeter, at 10% moisture content, relatively low. But in wet soil, there was a lot of attachment on the nematode. And this was pretty interesting for me because some literature said that the nematodes cannot move in water. They need a tiny film to be able to move. But this proved differently, that even in moist soil, they can still find their way and infect the insect. And this is important because these isolates were collected both from the northern part of Nigeria, which is known to be mostly dry, and the southern part, the southern humid part. So this is quite interesting that this can work in a humid region as well, where the spraying of chemicals might not really work because the interaction with water, which reduces the potency of the chemical. But on the leaf surface, it wasn't as much as that. And what this tells us is that spraying the entomopathogenic nematode on the leaves might not really work, but at maturity, when the fall armyworm drops to the soil, that is the perfect spot where the entomopathogenic nematode can target the pest and kill it. And what it does is that it causes the life cycle for the next generation. So this is pretty much what I explained previously in the other slide, for the sake of time. So what these two models tells us is that the amount of data we collect to monitor the temperature and that of the foraging and resilience, when we match them together, what it tells us is that with this set of data, we can find the ecological fitness of these nematodes. We can select the isolates for each region. And there, we don't use a single nematode as a blanket solution for every environment or for every insect, but we can select an isolate that works best for a certain region for a particular pest problem. And what is the next stage? Well, this is just one part of it, and it's mostly done in the laboratory. So we want to try out how these isolates will perform in the field. Will they be resilient to real stress factor on the field, and possibly combine these isolates to get the best of each of the isolates into one more or less super isolate that can perform ultimately well as a biocontrol agent. This is just a bit of the work, as you can tell, it's quite stressful. And honestly, most times I only see worms when I close my eyes. But at the end, I'm able to smile because I'm almost rounding up my work. And this was made possible by the support that I got from my supervisory team and from the funding agency. Thank you very much. If you'd like to reach out to me. Yeah.
Do you have a question associated? Now, my question is how UV stable is a nematode?
How what?
How stable to UV light? So sunlight, when you apply nematode, how do you formulate this endomorphagus nematode? Is there information around this?
Yeah, there are information about them. There are commercially available entomopathogenic nematodes. In Europe and some parts of Africa, in South Africa, I know, of course, in Senegal and in Kenya. But of course, the nematodes work best in the area where they are fully adapted for. So little room for importation, best used to develop the isolates for that region. And that is where hopefully we are getting to.
Thank you so much for your wonderful presentation of endomorphagus nematodes. Now on our last speaker, Amashit, enlightening us about cassava research and keeping the time.
Okay, I'll try my best. Okay, thank you so much. So actually, I'm a scientist based in Germany, but my heart is with cassava and I have an African heart. So today, I will show you these nice plants. This nice plant, actually, it's a food for 1 million people through the world. However, there is an invisible enemy. It's a cassava brown streak disease. It's turning this tuber, the healthy tuber, into inedible tubers, transforming lives and threatening the food security. So today I would like to guide you through this and show you how we try to find resistant and also map this resistant. So before I go in, just to show you where cassava is cultivated through the world. Actually, it's everywhere from Asia to South America to Africa, with the biggest producer Nigeria and biggest exporter is Thailand. So this is how cassava healthy looks like. I all the time like to start with this beautiful slide. So it's cassava, healthy cassava. It's not cannabis, so we cannot smoke it. So this is how healthy cassava looks like. However, wherever cassavas grow in the world, and this is the scope of my research, it's have a challenge. This is a virus disease, this is a fungal disease, phytoplasma, as you see, it's diverse. Today, I would like to focus on 2 diseases, is cassava brown streak disease and cassava mosaic disease. So cassava mosaic disease, actually, you can see it wherever cassava is grown in Africa, and it belongs to 11 species from the Gymniviridae and bigomoviruses. They all cause the same effect on the plant. They are different in the severity, however, they cause the same impact. So they make stunting on the plants, they cause curling on the leaves, the impact on the tubers by reducing the yield. So we find already resistant against this disease. However, the other disease that we don't, still a challenge for the breeder in Africa. This is a cassava brown streak disease. We see it in Eastern and Central and South Africa, and where we have an outbreak. It's Ebomoviruses, and it's belonged to 2 species, cassava brown streak and Ugandan cassava brown streak. They also have the same impact on the plants. So you see that they have yellow blotches or leaf chlorosis. However, the main impact, it's in the tuber, which because of the necrosis, it can reach about 100% losses from the yield. So cassava brown streak disease, actually, it's still a problem for the Africa, and it's threat to cassava cultivation in Africa, and for that, resistance is needed. Why it's needed? Because as you see in the lower pictures, it is depend on the livelihood of these farmers, where they can use this plant for foods. So this is what we heard in the sessions before, the scaling and the implement of the reasons are the solution. This is what we need because we need to support this woman farmer. So how we could go through this solution? We need to find resistant. So for that, we need to have a fast-forward screening workflow to screen for resistant. So this resistant, it's based on innovation, and it has different pipelines. So the first, that we need to know where to screen. And we went to the original sources of cassava and the South American germplasm, where we said, now we can take the chance and try to find a resistant. Second, we try to support the seed system. And for that, we have a propagation methods. It's called cut, root, and grow, where we can implement it in any place in Africa because it doesn't need a tissue culture facility. Third, we need to know the precise method of infection. So we can't rely on the vectors, and we can't rely on the whitefly to infect our plant because we can't know the time and we can't be precise. So our method was a grafting, so where we infected the plant with the infected material. Third, what we have to know, we need to know for what we are screening. Cassava brown streak disease, as I said before, it's caused by two species, cassava brown streak and Ugandan cassava brown streak. When we do this, and we know for what we are screening, we know what we are screening for. For that, we start with a specific isolate. So we use this pipeline where we screen for cassava brown streak disease, and we included cassava mosaic disease because we want to reach a dual resistant, a candidate where it has a resistance for CBSD and CMD. Because this plant at the end will go to the African field, and it has to have the resistance for the T disease. So we were able to find resistant, and I would like to convince you that the resistant already in the field. And with our partners through the world from Tanzania, Malawi, Vietnam, and DRC, and more, Mozambique also, it's on the list, and Zambia. But as you see, this is one of the prototype. It's the DSC493, a cassava progeny from South American plant that have a resistance for CBSD and CMD, and it's stable under hotspot in these places where we have a high virus pressure. So it's not about only science, it's about turning science into impact, like we heard yesterday from Dr. here, our professor. So we need to take this and put it into also impact, to put it on the breeder hands. So we transfer our technologies and we put it also with the farmer and the breeder, and they were able to screen, and this is our success story from IITA DRC and IITA Antari, Tanzania. So now we find the resistant, we know the resistant stable in the field, but the question why this plant is resistant. So we need to map the genes for today to secure the healthy plant for tomorrow. So this goes from viruses and also from the plant. So I look for resistance from the two sides. From the virus side, when you ask about resistant, you question two things. You question the replications, and here was our questions. Does the virus replicate in a resistant plant? And as you see here, the virus was barely detectable in a resistant plant. So the virus is not able to replicate in a resistant plant. When we ask about the virus movement, the question, can the virus move inside the plant? And here we have a highly sensitive technique called RNA scope. And with this technique, we were able to localize the virus inside the cutting from cassava tissue. And this is a cassava tissue, as we see here, from the left to the right, highly resistant to highly susceptible, the red reflecting the virus inside. So you see in the susceptible plant, we have a lot of red dots or cluster reflecting by a lot of viruses. While in the resistant, it was not, we were barely able to detect the virus. And when we detect the virus, we try to be specify exactly to know where this virus is. And we used a green fluorescence protein antibody to localize the cell. And as you see, the CF element here, it's the phloem cells, it's in green, while the companion cells where the virus is the red. So we know that here the mechanism of resistance from the virus side is restriction in the virus replication and restriction to the virus movement to the phloem companion cells. And here comes the question, why the virus is restricted to the phloem companion cell? How come that after I put my virus inside the plant, and I'm sure that it's in, so it's moving through the phloem because it moves with a sink source. So the virus should be there. It reaches to the companion cells, however, it's not able to go outside. So for that, I need tool, and my tool was infectious virus, cDNA and clone, and also GFP tagged clone. So we used this clone and we said, okay, from where I should start, actually. I have a lot of genes, what I should tackle. So the most important things and most unique things about cassava brown streak virus is the HAM genes. The HAM genes upstream the coat protein. This is make it unique for other viruses because it's very unique that other viruses in the whole families doesn't have these genes. So this was a start for our work, and we asked maybe the HAM then has something special, make the virus localized to the phloem and not able the virus to move to the companion, to the parenchyma cell to replicate. So we use this GFP clones and we agroinfiltrated the pentamiana, as you see here in the pictures, and you see the pictures down. This is the plant then. It's a video, but it's not seeing. So you see that the green was in the pre-nuclear compartment and the plasmodesmata and plasma membrane. So I didn't see anything specific about the HAM. And while I thought that I can localize to the plasmodesmata, which is the gate for the cell-to-cell movement, it was not. So I said, okay, I'm working with model host. I should try this in cassava. In cassava, the agroinfiltration doesn't work. So we have to use another technique, which is in our cases here, it's a bombarding, uses the gene gang. And then we delivered our clone inside the cassava leaves, and I used susceptible and resistant, as you see. However, in both cases, I was not expecting what I see. So the protein was localized mostly to all cell compartment, pre-nuclear compartment, plasmodesmata, and plasma membrane. So no specific localization. I said then, okay, maybe it's not the HAM alone, it's the HAM with another proteins. So we tried all the several protein, the combination to co-express those two genes. However, this is only an example. We didn't see any effect on this co-localization. So the outlook now from this work that maybe it's not 2-player in the game, maybe it's 3-player. So now we want to add more protein to study the protein-protein interaction to see how this protein localized, because for sure there's something that from the virus side prevent the virus from moving from to the other cell. But this is from the virus side. So let us move to the plant side and see what's happening. So before I go to the plant side and say I'm searching or mining for resistant genes, first I need to have a reference genome from my resistant plant, because all the reference genome is the database highly susceptible plant. So if I want to map anything against my resistance, I'll maybe miss it. So this was the idea of Stefan, where we said we need a reference genome from our Col40s, our golden line, the high resistant line for CBST. And we were able to find this. So it's published now. So it's our baseline. So the next, then we said, okay, now we have the genomes. The next, let us look more deeper and check for if we could find a QTL, because we screened a lot of crosses. We didn't only screen germplasm, so we screened a lot of crosses, and this was also with a collaboration with a breeder through the world where they produce these crosses. So we did a precise phenotyping for these viruses. And our collaboration from CIAT, she did the genotyping, and we were able to reach a 3-cask marker that are related to the resistant. And now we are in the validation step for this marker. So this is from one side. But we didn't stop here, and we said, no, maybe there's something else. So what we know that CBSD is a quantitative trait, and it's dominant. So we found it in the first generation. But what we know also that the CBSV resistant is different from the UCBSV resistant. So we will not stop only to find this, because for sure different genes also responsible for resistance. So we said, okay, what about the early response of the plant? And for that, we did the transcriptomic analysis. And what we see here quickly, that we find a highly significant result in the first day after grafting. So the resistant really shows highly. Then what we did also, we looked for the small RNA and said, maybe we can find also a hint in the microRNA, which we also find. And this was something lead us to lipid binding protein, which now we are taking the lead. We didn't stop here. We said, okay, I want to go more on the proteomic level. And this is what we did actually with our collaboration in the Thailand. And we also find it's a lot of protein, but what also comes here is a lipid binding protein. So now I have also a candidate. So just to come and conclude everything what we have seen at now. So we looked for the viruses, we looked for the genes of the viruses, for the protein of the viruses, for the localization. We looked on the plant, we looked on the DNA, which tell us what is possible. We looked on the transcript, which tell us what is the genes that is already the transcript or expressed. We looked on the protein, which is the functional. But actually what we want to look, and it's what we want really to prove it, is to look on the protein interaction and to look on the metabolites, because this is the final step of a phenotype. So instead of going from genotypes to resistant, I want to go from the phenotype, from what I have, and go back. So at the end, so I reach my end slide, and I want to say that mapping resistant in cassava is more than protecting a single crop, actually. It's a securing food system, livelihood, and health. So my home message is saving a plant actually can saving a life, and it's not about science and it's not about research. It's about real solution for real people. Thank you so much. I made it up on time.
Thank you so much for speaking first about impact, so that you have delivered the plants to Africa, and then combining fundamental research on this very complicated crop with practical solutions. So this presentation is up for discussion.
Sama, this is brilliant. I really enjoyed the talk, even though I wish you could have continued for more time, if time was there. This is just out of curiosity. It has nothing to do with the presentation as such, or the topic of the presentation. The leaves is very bad when they are affected by the virus. Is it a possibility that this could jump to human?
To human?
Yeah, in norovirus, we know that there are possibilities in terms of food safety of cassava leaves are consumed in Congo a lot and all that.
Yes.
Is there that remote risk of a jump in terms of food safety aspect of?
Yeah. I think for our viruses that we are working with, there's no way that it can jump to the viruses. But if you analyze the blood of Stefan, you will see a lot of antibody against plant viruses. So actually, we could infect ourselves somehow, but it will not. If we are not in a health risk, Yeah. For that, I don't see any risk that this can jump from plant to human.
How many million Africans and elsewhere have eaten cassava leaves and are all
infected with this?
or sick from something else?
Actually, you know that in Africa and in Congo especially, where we go, when we went to the field and the woman farmer, they collect the infected leaf because they like it more, because they are more sweet. So actually, what they eat, they are eat more the infected than the resistant one.
Like eating locusts.
Yes.
Sama, thank you very much.
Thank you. Thank you.
For the sake of time, I herewith conclude this session. Thank you very much to the speakers. Dr. Kasim, for your inspiring introductory to molecular biology and insect control. To all of you to provide us with pest risk assessment, invasive species, and so on. So I'm very happy and thankful for the ones who contributed to the discussion, and I herewith close the session because we have another poster session to attend. Thank you so much. Thanks. Oh.
You in the meeting, I want to use my voice and use my let me see. But I have everything that I don't have to there is empty space. We just record it.
You want to record here?
Yeah, so I want you to close the Zoom. I will open Zoom on my